Hey everyone, welcome to the second edition of the Neural Ensembles meeting that Rafael Juste and I started last year. It was really wonderful meeting last year. This year is going to be the continuation of this meeting. This time we also have Rosa Cossar joining the team of organizers. And this year we'll be having about 1,200 people joining this online meeting, which I think express, in my opinion, great success. So just briefly, what are neuronal ensembles? Rafa asked us briefly define from our perspective. The way I define neural ensembles is essentially the group of coordinated neurons whose activity is coordinated through the internal dynamics and recurrent connectivity of the network. This is my definition. And this year, I'm quite excited about the meeting because we will be really going through the evolutionary tree from animals, from mammals to animals essentially without a centralized nervous system. And then we will see different implications of the use of ensembles. So I'll pass the word to Rosa now, also just to say a few words. Please, Rosa, go ahead.
I unmute. Thanks. So I'm very excited to be part of this, the panel of organizers. So it's a great honor to be here today. I wanted to, so I have a developmental point of view on neuronal ensembles, and the questions we ask are they predetermined? What are the mechanisms by which these ensembles form? I think those are questions we may be able to address throughout this meeting. My definition of ensembles is, I mean, it's similar to yours. It's the idea. First, I wanted to say is that it's a term that we use a lot, and we don't have really a standard or precise definition. And I would say there are groups of locally or internally correlated neurons that make a functional unit in the sense that they are reactivated for a given brain function. That would be my definition today of ensembles. Yeah, so Rafa.
All right. Thank you, Rosa. Hello, everyone. Either whatever, good morning, good afternoon, or good evening, wherever you are in the world. We know we have people from all over the world. And I wanted to welcome in the name of the Neurotechnology Center. So this is a center at Columbia University that was created in 2014 as Columbia's answer or response to the US Brain Initiative of President Obama. And the purpose of this center is to promote research, outreach, and advocacy of neurotechnology. So this is defined as methods to record or activate neuronal and circuits and glia, of course, as well. And part of what we've been doing since 2014 is host international meetings on topics that have to do either with the development of technology, on the application of technology, or on ethical and societal consequences of technology. We've done a lot of meetings. In fact, I'm proud to tell you this is number 15. And when COVID came, we moved online. And this seems to have been a very successful move because we've been able to reach and spread information to people all over the world for free, a much larger cohorts of people. So, and I wanted to tell you that this year, this meeting is sponsored by the Chen Foundation, the TCCI Foundation, and we're thankful for their support. And at the end of the meeting, we'll have the director, Yan Li, the director of the Chen Foundation, will say a few words to close the meeting. So what is a neuronal ensemble for me? So everyone may have a different angle, and this is part of the question that this meeting hopes to answer, at least to provide people with the rich phenomenology and concept behind this idea. No? So for me, I resonate a lot with what you've heard. For me, a neuronal ensemble is a group of neurons that is firing together, is coactive. And of course, that depends on the window of time in which you define this coactivity. But we think, and I'll show you data, that there's such a thing as a coactive group of neurons, that the activity in the brain is not like all percolating randomly, that there's organized. No? And this activity, this coactive group of neurons repeat, and they happen spontaneously. And as you've heard from Rosa, they are functional units. So they have either a behavioral significance or a functional significant. No? So to me, neural ensembles are modules of neural circuits. No? And the idea of neural ensembles is not new. In fact, it's over 100 years old. But one problem is that investigators have used different names to talk about these things. No? So, and sometimes unclear if they're talking about exactly the same thing. No? But if you go back 100 years ago, already in the Cajal lab, Lorente was talking about what he called chains of neurons, which meant something very similar. So we currently connected the chains of neurons that would have spontaneous activity. No? Hebb introduced the concept of assembly, a neural assembly. So this is very, very similar to neuronal ensembles. And the literature is also divided. Some people use the term ensemble, some people use the term assembly. We chose the term ensemble for this meeting because Sherrington coined the term ensemble. That was before Lorente and before Hebb. And also results from my lab that are now in press in eLife show that these ensembles are non-Hebbian. They don't rely on Hebbian plasticity. So that would be a reason not to use maybe the word assembly. But other people have used words like attractors, pumps, etc. So the literature is a little bit confusing in terms of the nomenclature. And just to finish, why ensembles important? Why do we care about ensembles? So I would argue that how neuronal activity is translated in thought, maybe to me, the most important question in neuroscience. No? How do you go from a bunch of neurons getting activated to mental cognition? No? And how do you build a thought? What is a thought? No? And for me, and for many of us, the critical level is the neural circuit level. So obviously the nervous system is built from molecules, to synapse, to neurons, to neural circuits, to systems, to the whole brain. No? And of course, everything is important. But if you want to go for this question, what is exactly a thought? My intuition is that it's the neural circuits where it's at, and you need modules. So these ensembles could be modules that could be used by the brain to symbolize things. So the meeting is focusing on ensembles. We're going to start talking about methods. The reason is because methods go hand in hand with scientific discoveries. And the reason why ensembles are important now and not 50 years ago is because now we have methods to look at groups of neurons firing together, and we didn't have that earlier. But as you heard, we're also going to be talking about two groups of animals, so to be more precise, of metazoans, cnidarians and bilaterians. And some of you may have no idea what are we talking about, a cnidarian or bilaterian. So you will hopefully learn that by the end of the meeting. But cnidarians have the most primitive nervous systems. Bilaterians, animals with bilateral symmetry like ourselves, have more sophisticated nervous systems. And there's actually good evidence for ensembles across all the animal kingdom of metazoans that have nervous systems. So this would be very interesting to provide this evolutionary angle. And with that, we're going to get started. The rules of the game: the speakers have 25 minutes to speak, and there will be a 5-minute Q&A. The audience can send questions through the Q&A button in the Zoom interface. The other speakers could also send questions either by raising their hand or by typing questions in the chat. And at the end of each session, we're going to have a panel discussion with 15 minutes where we're going to bring all the speakers and essentially have them interact with each other and ask them questions. So with that, we're going to get started. The method session, the first speaker is Misha Arens from HHMI Janelia. Misha was invited because he's doing beautiful work, some of the most beautiful work using optical methods to image and decipher the function of these ensembles in zebrafish. The title of his talk is "A Positional Integrator in the Vertebrate Hindbrain." Misha, take it from here.
Hi, thank you, Rafa, Rosa, Emre, and also Lux. So very pleased to be part of this meeting. I just missed, is it 25 minutes or 30 minutes? 25. Okay.
You have 25 to speak and then 5 minutes of questions.
Yeah. All right. So I won't ask whether people can see my slide.
Yes,
right?
it's
But
perfect.
let me know if you can't. Okay, good.
Yeah, we can hear you and see your slides, so you're good to go. Yeah.
Great. So yeah, thanks again for having me, and I look forward to a great meeting with all the speakers, and hopefully also some great discussions over Zoom. So I would like to speak about a positional integrator that we discovered in the hindbrain of larval zebrafish. So in the lab, we're interested in brain function, eventually human brain function, but the human brain is hard to access. So we use other models to ask, how do large populations of neurons collaborate to implement the computations underlying flexible behavior? And you can see that the concept of ensembles is embedded in this, because in the end, it's all about large populations of neurons communicating with one another. And some of the many functions of the brain include learning, motor learning, and learning about the physics of the environment, navigation through the environment, and memories of where animals have been and what they've experienced there, and also the control and sensing of organ systems. And I think it's remarkable that this one organ in the body of animals can do all of these things often at the same time. One way in which it can perform all these functions at the same time is because it's densely connected. So neural processing is distributed, and networks subserving all these different functions are connected to one another, and in some sense aware of what all the others are doing, if I can anthropomorphize. So neural processing is intrinsically distributed. Any given behavior tends to be mediated by multiple neurons, and that are connected, and that also induce plasticity across multiple connections. So in this talk, I would like to present to you work that is organized in the way we usually tackle biological questions, which is to consider the goal of the system, or the goal of the animal, the network, the algorithm that the network uses, and the biological implementation of that algorithm. And I'm going to talk about a behavior that we call positional homeostasis. And this is work spearheaded by En Young, who's an extremely talented and creative postdoc in the lab, in collaboration with other lab members and collaborators, Martin Swart, Mikhail Rubinov, Zicheng Wei, Benjamin James, James Fitzgerald, and others. So let's start with the behavior. This is a fish. This is not a zebrafish. This is just a fish swimming in the river nearby Janelia. And it's performing a behavior that many fish perform, which is it swims against the water flow. And in this way, it can stabilize its location in space, even though it would normally be perturbed by the flow. If it wouldn't do this, it could end up in dangerous locations, or if you imagine a body of water draining, it could end up on dry land. So this is an important behavior for fish with a clear ethological interpretation. And the common way to describe this is simply the fish swim against the flow. They see the visual world move around them, or perhaps they sense some water flowing around them. But typically, this is a visually driven behavior. They see the world move and they swim in the opposite direction. So that's one interpretation of this behavior. Another interpretation is that they have an explicit notion of where they are in their environment, or at least how much they've been moved by the current in the past minute or so. And so the first thing we did was to try and test, to try and distinguish these two possibilities. And to be more formal about it, we considered one of these possibilities by analogy of place cells, where brain areas like the hippocampus and the entorhinal cortex have an explicit representation of where the animal is in the environment. And we asked, what could this look like from a control theoretic perspective? So just to be clear, we were not studying the equivalent of the fish hippocampus. We looked across the entire brain. We tried to distinguish these two models, these two control systems. In the first one, we imagine that the flow velocity induces a velocity of the visual field of the fish, and that makes the fish swim in a certain direction with a certain vigor. The way we look, we started looking for cells that might represent this control system is to use VR behavior. So we embed the animal in Agoro, so its head doesn't move, and we can manipulate its environment and also close the loop between behavior and the fish's swimming to mimic the visual input that an animal would receive if it were actually swimming in a real environment. And so we use an environment with a semi-random background through which the fish can swim. So this is an environment that has a lot of flow cues, but no explicit landmarks. And we mimic the water flow virtually by presenting visual flow to the animal in the opposite direction. So I'm going to show you this as if the animal is moving through real space, but keep in mind, this is all through virtual reality space. So let's introduce this dashed line for our own reference. If we switch on the virtual water flow, the fish will move and it will swim. And if this system is tuned kind of correctly, the animal will approximately stay in the same location. But if you perturb the animal ahead of time, so represented by this hand, and in VR, this is simply implemented by a movement of the visual stimulus, and then wait a couple of seconds. If you would then switch on the virtual water flow, the animal would perform the same movements and end up in a different location from where it started. So this is that simple control system where the animal is responding to the instantaneous stimulus input. And so we get these two trajectories which don't converge in these two different trial types. We can interleave those trial types and compare their averages. And this separation is a signature that the animal has no memory of what happened before. If we contrast this now to a control system, which does have an explicit representation of the displacement that the animal has experienced in the past, which is the integral of the velocity that the animal has seen in the past, then you'd get something different. So in this case, if you displace the animal ahead of time, wait a couple of seconds, and then switch on the virtual water flow, the animal would do something different. Perhaps it would wait longer before it starts swimming to get back to its original location. And this should also work the other way. If you pull the fish backward, it might swim a little more vigorously or a little more often to get back to where it was before. And so these convergent trajectories on these different trial types are a signature of a positional memory at the behavioral level. And what's important here is that we loop, so every trial starts at the end of the swim period of the previous trial, so that the animal can choose where it starts and can decide during the swim period whether or not to return there. And so we call these the pre-displacement period, where we forcibly move the animal through the environment, the delay period, where the animal just hangs out for a few seconds, 5 seconds, 10 seconds or longer, and then the swim period, where it swims against this virtual current, and by balancing the backward motion and the amount of forward swimming, the fish can decide itself where it ends up. Now we can simulate these two control systems and ask what does the control system do on these different trial types. And on the left, you can see that this control system that doesn't have a memory does not cause these trajectories to converge. And on the right, you can see that control systems with a memory do produce converging trajectories. We can now ask, what does the fish do? And it turns out the fish looks very much like the control system on the right with a memory. And this memory, actually, if you simulate it, has a very long time constant. You could see that averaged over 8 fish, but also an individual fish, they make these trajectories converge. And what we've done here is to center it on the middle trajectory to make the comparison across fish easier. So this looks very much like the control system with an explicit displacement memory. And this displacement memory is what we were really interested in. So we wanted to figure out where it is. But first, we wanted to look more carefully at the behavior to see whether this was some peculiarity of that particular paradigm or not. And it turns out that they have this displacement memory in all kinds of situations. For example, this here is an example fish, trial averaged, where the fish was pushed forward in virtual space over varying distances, and in each case manages to get back to where it started. So if you compare the initial point to the final point, you can see that at the end of the swimming period, the fish comes back to where it was before. And you can also do this in a much more complex situation where you randomly move the fish forward or backward over a period of 5 seconds, then wait 5 seconds, and then divide up the swim period into 2 and hold the fish constant in between, so that the fish has to keep the location in memory for almost half a minute, and it can still do it under these much harder conditions. You can see that here across the fish population that we tested. It's still quite good at bringing the self-location back to the starting point. So all this means that we're starting to take this control system analogy seriously. And as I said, if you simulate it, then trajectory convergence requires a displacement memory with a pretty long time constant. And so to look for displacement memory in the brains of zebrafish at single cell resolution, we now turn to methods that we and others have developed over the past few years, where we combine VR behavior with light sheet imaging to record from the entire brain during virtual reality behavior. And in this way, we can get whole brain data sets where we optically section the brain into about 30 or 40 planes. Here you can see an example recording where these are entire brain is imaged more than 3 times per second. Every cell is imaged more than 3 times per second during behavior. This is during a couple of swim bouts, and we retain single cell resolution throughout almost all the brain. So we record from about 120,000 neurons and also about 20,000 glia. So this is a way to really bridge scales and look at both the cellular scale, the network scale, and the whole brain scale. And we were explicitly looking for this displacement term, the neural representation of this displacement term. So we did this during VR behavior. We used online large-scale data analysis methods that we developed and implemented on the cluster within Janelia Research Campus, and analyzed this whole brain activity for representations, cellular representations of where the fish is in space. And one way to do this is to look at the correlation between the fish location and neuronal activity. And if you do this, you get a functional brain map or a brain activity map of position and coding cells. This is over multiple fish combined. And this gives you specific brain regions on which we can subsequently focus. And this includes an area that we call SLOMO, for self-location encoding medulla oblongata neurons. We have an idea of what this brain region is homologous to in rodents, and I'll get back to that later on, or in mammals, I should say. And then the inferior olive shows up in this map, the cerebellum. These are known to be connected, and then also some other brain regions, including the dorsal raphe nucleus and the habenula. Now, you can look at this data in multiple ways. So this is one way, position encoding cells. In other ways we looked at this data was to ask for the memory of pre-displacements, and also to ask whether we can predict future swimming behavior from brain activity, given that the future swimming behavior is determined by the past displacements that the animal has experienced. So you can look at these data in all different ways, but the resulting brain maps are very consistent in highlighting, this specific set of brain regions. Now, it's already known that the inferior olive and the cerebellum are connected, so we asked how does SLOMO fit into this connectivity picture. So we worked with the Engert, Lichtman, and Jane groups to do reconstruction of SLOMO cells. This was done by Martin Swart. And we found that these SLOMO cells have axons that project to the field of dendrites of the inferior olive. We also found that the slow-mo cells are GABAergic. So this is now an inverted color map where the black cells mean overlap between slow-mo neurons that are functionally identified and GABAergic neurons, which are identified molecularly. So this is a group of GABAergic neurons, which projects to the inferior olive. And now we can start hypothesizing a brain-wide network that implements this behavior, where the slow-mo to the inferior olive connection we've established, not only through EM, but also through light tracing and optogenetics. Inferior olive to cerebellum is hypothesized, is known, and the other connections are hypothesized and still need to be fleshed out. But let's start to look at this initially upstream region of the inferior olive area slow-mo and ask what does activity here look like as animals move through virtual environments. So here's an example fish where the cells are color-coded according to whether cells are activated more when the fish is moved forward or when the fish is moved backward. And here I should say that this diagram shows an oversized fish. The actual amount of displacement is almost 10 times the body length of the fish. So in this data, there's substantial movement through the VR environment. And here's now one example cell that increases its activity when the fish moves forward and decreases its activity when the fish moves backward. And another fish with an inverted activity pattern where increases in cell activity correlate with backward physical motion through VR. And in each of these cases, remember that in the yellow period, the pre-displacement period, that's where the animal sees visual motion. And in the white period, the animal sees a static stimulus. So this means that these cells have persistent activity encoding what happened in the past. And this is why this represents positional integration. Now, cell 2 is pretty easy to interpret. You flip the y-axis and you get a fairly good representation of where the animal is in space. Cell 1 is harder to interpret because it has this ramping activity, which shows even when the fish isn't moved in black. And this ramping activity encodes other variables that I'm happy to discuss later. But despite this complexity of encoding, we can look at the dimensionality reduced version of this data at the network level. So these are several hundred cells embedded in 3 dimensions through principal component analysis. We can basically read off where the fish is in space. For example, when the fish moves backward and back through PCA space, the fish moves along this trajectory. So we think we can decode it, but we can actually check this, whether we can decode the fish location. We can check this through neural decoding, where we used a very simple decoder, a simple linear weighting of the activity of each neuron to estimate where the fish is in space. And here you can see that being done during the experiment where we randomly wiggled the fish forward and backward in space and then let the fish swim back if it wanted to. And these cells are very informative about where the fish is in space. Now, the cells don't only encode forward and backward motion. Here you can see that they encode forward and backward motion during the complex trajectory that I just showed you. This is an example cell, but this brain region also encodes sideways motion. As you can see here, the fish has moved sideways through the VR environment, and there are cells in this region that encode the amount of sideways motion, also in a persistent way with a very long time constant. Okay, so this was what I showed you here was all correlational. It's about how these cells encode the movement of the animal through space and how they have a memory of where the animal is in space. But we would like to get into mechanistic detail and a mechanistic understanding. So for that, we used our interactive platform where we can image, analyze, and then perturb at single cell resolution. So we can create these brain activity maps through various analysis methods, and then go in with the experimenter can go in and perturb this system anywhere in the brain using 2-photon random access cell perturbation. And so we asked whether slow-mo is necessary for this displacement memory. And first, if we look at the control system by analogy, this is the intact control system that makes the trajectories converge after displacement. If we now remove this displacement term after ablation, we get the control system without the memory that you also saw before. So what happens now if we do this to the slow-mo region in the fish? This is behavior before ablation. If we take out the slow-mo region, which we've hypothesized is the integrator, then the fish does that. It still swims against the water flow or the simulated water flow, but the trajectories no longer converge, exactly like the control system that lacks the memory. So this is a validation of the hypothesis that slow-mo contains the memory, but we can go further and we can ask, what if we ablate part of slow-mo? So if this is really a network that implements the memory, and this again comes back to the neural ensembles where neurons work together in a network, if we only ablate half of it, which can be either the cells that represent the backward displacement of the fish or the cells that represent the forward displacement of the fish, then afterwards, the memory capacity of the entire network is gone, even though half of the cells are still intact. What's interesting is that the cells that are intact are still responsive to the stimulus, as you can see here at the bottom. They still respond to visual flow, but they lack this persistence. They lack the memory of that translates visual flow into positional change. So this suggests that recurrent connectivity within slow-mo supports the long time constants of memory. And as I said, this comes back to ensembles and attractors. Now, the opposite question is, if we manipulate this activity, can we in some sense implant memories? So that's what we did using a system, again, the system where we do functional imaging first, analysis of brain activity to figure out which cells encode the displacement, and then manipulation, this time by optogenetic activation. And if we activate either the cells that encode backward movement or cells that encode the forward movement, we can mimic the future behavior that the animal would have performed if it had actually seen backward or forward displacement. You can see that here. For example, if you look at the green cells, this stimulates backward neurons. The fish will swim more as if the fish had actually been displaced backward in space. I animated this. The behavior is consistent with the fish having actually experienced these displacements. Okay, so both of these establish slow-mo as the site of positional integration, and it was a surprise that this occurs in the brainstem. Now we can look further in the network. We know that slow-mo connects to the inferior olive, so let's look at what inferior olive cells are doing. First, we found that inferior olive neurons also encode backward and forward displacement in a way that's consistent with the way they're wired up to area slow-mo. So backward displacement encoding slow-mo cells project to the medial part of the inferior olive that represents forward displacement. And that's consistent because slow-mo cells are GABAergic, so they're inhibitory. So you would expect the opposite tuning in the inferior olive. And the cells in area slow-mo that represent forward displacements project to the lateral sides of the inferior olive, and those lateral sides of the inferior olive represent backward displacements. And again, that's consistent because of the inhibitory nature of the slow-mo cells. And if you look in the inferior olive, again, this is centered data. If you simply subtract the medial and the lateral activity of the inferior olive, you get a pretty good representation of the displacement of the fish. And we interpret this as a positional error signal, which is a novel type of error signal that represents the positional error in of the animal as it's trying to stabilize its location in space. And this has extremely long time constants of tens of seconds. You can use this inferior olive activity to predict what the animal is going to do in the future. So here you can see an experiment where N took activity in the inferior olive at the very start of the swim period before the animal has performed any swim bouts, and she can predict when the animal is going to swim in the future. And so finally, to check whether this is all mechanistically sound, we ablated the inferior olive and compared the behavior before ablation to the behavior after ablation, and found that again, the fish was unable to make the trajectories converge. That means it's still performing the kind of memoryless optomotor response. It's still swimming against the flow in the direction of the visual flow, but it's unable to encode and respond to the memory of displacement. Or actually, we know that the slow-mo still encodes the memory, but the fish cannot respond to it because the route via the inferior olive to behavior is now broken. So finally, we think that slow-mo overlaps with the nucleus prepositus hypoglossy, a brain region commonly defined in mammals, not conclusively identified in fish, but through a lot of literature search and comparing atlases and cell types and connectivity, we think that this might be the region to search for these kinds of cells in mammals, and we hope that, that this will happen. So again, we're looking at multiple levels, the goal of the animal and the goal of the system, the algorithm used, and the neural implementation. And through this multi-regional and ensemble investigation, we believe we can really start to connect these control systems to the multi-regional, multicellular implementation. Of course, there's still a ways to go. We have EM connectivity data that would now allow for a much finer investigation of how these cells are connected within and across regions. Okay, so with that, I would like to again thank Anh Young, an amazing postdoc in the lab who's done all this work over the past few years, and her collaborators, Martin Schwartz, Mikhail Rubinov, Xicheng Wei, Benjamin James, and our long-term collaborator, Janelia James Fitzgerald, and all the other people who've been in the lab, collaborators and labs that we've had the privilege to work with. Thank you so much.
Thank you, Misha. This was really beautiful. We have a question from Henry, and I remind all the attendees that they can send questions by typing into the Q&A box. Henry, you can go for it.
Fantastic, Misha. So these cells are GABergic, that you see this persistent activity, and then you ablate some, and then you get rid of the persistent activity. Can you say a little bit more about how you generate persistent activity with a GABergic network? How do you, I mean, can you speculate a bit about this?
Yeah, it was initially counterintuitive, but we've been working with Xicheng Wei and James Fitzgerald on models that can produce this. So one way to create a model with this property is if cells have a spontaneous firing rate and they inhibit each other in that way, that they suppress each other's firing rate in a recurrent manner and produce long time constants. Another way is what I think is a very interesting possibility is that there are rebound currents in rebound channels in these cells, so that inhibition has a future or simultaneous excitatory effect as well. And in this way, you can imagine one cell kind of transiently inhibiting another, but causing an increase in the probability of firing right after this inhibition. And this sort of can also accumulate within neurons. So some of these channels have quite long time constants, so up to a second or so. But these time constants can then propagate through the network to really produce quite long time constants across the network. And we see some signatures of connectivity as well, where there are some cells with shorter time constants and some cells with longer time constants. And you can also recreate this through network models that, again, now Benjamin James Fitzgerald, and Xicheng Wei have been working on, where specific connectivity together with this rebound property produces this range of time constants in the network.
Thank you. We have a question from Rosa. Rosa, you want to unmute yourself and go for it?
Thank you. It was beautiful. Thanks. I have a question related to the size of neuronal ensembles. You said you're able to image 120,000 cells at the same time. And my question is, what's the fraction of cells that are involved in that, the representation of displacement? I mean, so they are, yeah, can you give us an idea of like the, either in absolute numbers or fraction of cells or?
So the number of cells in area SLOMO are typically a few hundred, maybe 300 or so. If you then consider the cells that they're connected to, of course, they're part of a multi-regional network. So in a recent experiment, we ablated the inferior olive and imaged SLOMO. SLOMO is still functional. So it seems to be able to encode this memory if you remove the connection to the other brain regions. And we did this because we were wondering whether there's some recurrent property that runs through multiple brain regions, maybe the inferior olive, cerebellum, and back, that endows the system with these long time constants. But it seems to be that SLOMO, with its connections to upstream regions, has sufficient, ensemble properties to be able to store these. But then, of course, if you consider the downstream regions and the networks that it interacts with, you could get a lot more. So it really depends then on kind of the criteria that you set for how do you define encoding, and how do you define as the signal gets closer to behavior, there's still traces of encoding. So the inferior olive still has this signal, then the cerebellum and premotor regions, they become closer and closer to actually motor output, but of course they have signatures of the displacement signal still. So then it depends on kind of your personal definition of how you call ensemble encoding. So you could get to thousands of cells very quickly once you broaden your view. So and I think that's the great thing about neuroscience.
Thank you, Misha. We have time for one quick question coming from the audience from Ron Yu. It says related. Have you tried to ablate random subsets of the neurons and see when do you lose the positional information in terms of percentage of cells?
That's a really interesting question too. We haven't. So I think, I think that would be a great experiment to do. All we've done is to take out one functionally defined population, either the forward encoding or the backward encoding cells, and looked at the remaining ones. But yeah, going cell by cell and figuring out the robustness of this network would be really informative, and would also connect very easily to models of this network.
Thank you. We're going to have to move on, but I encourage the audience to continue to send questions, because we'll have time at the end of the session for a panel discussion, and also at the end of the meeting. So, and the purpose is just to encourage interaction and inquiry. So thank you, Misha. We're going to move on to the next speaker, Valentina Emiliani from the Institut de la Vision in Paris. The reason she's invited has to do with developing methods to activate these neural ensembles with single cell precision. So you have to realize that these ensembles are not nicely tucked away in different parts of the brain, but they're all intermixed, as you've seen from Misha's beautiful data. No? So if you want to disentangle what they do, you need to be able to go in there and turn on these neurons, but not the neurons that are next to it. So this is not a trivial challenge. And Valentina is one of the pioneers developing holographic methods to do this. The title of her talk is Ultra-Fast Light Targeting for High Throughput Precise Control of Neuronal Networks. Valentina, you can take it from here.
Thank you. So good afternoon, good evening to everybody, and many thanks to Rosa, Emre, and Rafael for inviting me to join to this a very interesting workshop. So I think now the video is on. Is it right?
Perfect.
Okay.
Yeah.
Okay, thank you. So the topic of this talk will be more into giving you a sort of overview of a number of approaches that my lab and other have developed in the past year to use light to control the activity of neuronal circuit or neuronal ensemble by using optogenetics. And I will first give you a quick overview of a number of approaches that has been developed in the past years. And in the second part, I will show you a few unpublished data where we are pushing this approach even further to increase the number of achievable targets and increase the temporal resolution. So if we just start the story from the beginning, there have been already a number of experiments that has been done using optogenetics, and where the very first step was to try to develop approaches to perform a very precise mapping of brain functioning. And this was done at the beginning with relatively simple illumination method, which consisted in shining light on relatively large brain region, and then count on the specificity of genetic targeting so that it was possible to reach a precise correlation among the evoked response and the specific cell type that was activated. So this give to the community for the first time the technology to perform a precise mapping of brain functioning with cell type specificity. But as Raphael was mentioning in introduction, there is somehow a limitation in this approach, and this is that when you shine light in a specific way, eventually all the positive cells here represented in green are going to respond in a synchronous way, and we cannot really select which kind of cells we want to activate. While if we now look at the activity of circuits under physiological situation, this is just an example of the visual cortex, but we are familiar with many other circuits that have a similar behavior. What we can observe is that even genetically identical cells may have a completely divergent activity pattern. And so now if we want to enter in a phase where we start to mimic this kind of activity to try to find the role of a specific neuron ensemble, or even their size, or their reveal how important is a specific spatial temporal pattern produced by a specific neuron ensemble, we need somehow to transition from this whole region optogenetics to a more sophisticated combination of approaches that we termed a few years ago circuit optogenetics, meaning within a combination, with this a combination of technology that give us the capability to manipulate single or multiple target cellular resolution, to reach multiple targets with a millisecond temporal resolution and precision, and eventually control neurons organized in a three-dimensional structure, so light delivering at multiple planes. So I will first show a number of approaches that my labs and others have developed to fulfill these three requirements, and then we will go into the new part. So how can we manipulate a single or a group of ensemble of neurons with single cell resolution? This obviously requires to replace wide field, the single photon excitation with focus to photon excitation combined with holographic light shaping. So in this case, the key device is a spatial lay modulator that addressed with the specific phase holograms enable to redistribute the way the light propagates at the sample plane so that we can eventually reshape the light so to reproduce precisely any image template that we have drawn by computer. And so this can be used to enlarge the illumination shape, so to be more efficient into photon excitation, but also to multiplex the photostimulation on multiple targets. And so in this case, the same device can be also used to generate, for example, disk that can reach multiple targets simultaneously. And the same approach can also be used to generate simply multiple diffraction limited point that then with the use of a galvanometric system can also be scanned through into the different cell soma. And so in the first case, we speak about parallel holographic illumination. In the second case, about scanning holographic illumination. So both cases enable us to reach multiple targets with millisecond temporal scale. So in our lab, we were more interested in working on full parallel holographic illumination to optimize the temporal resolution and precision. So from now on, I will mainly concentrate on further implementation we propose for full parallel holographic illumination, but in the second part, most of what I will show is also compatible with spiral scanning. Okay, so parallel holographic illumination. We want to reach multiple targets, and when those multiple targets are close to each other, we also need to control the axial propagation of beams. So we propose together with computer-generated holography to combine the approach of temporal focusing with, in this case, the key elements is dispersive grading, which is plays on a plane conjugate with the objective focal plane. And so that when the light is going through the grating, these enable to increase the absorption of the light selectively at the focal plane. And this is something that can be easily understood if we directly look at what happens here. So this is an holographic beam that is made to produce an illumination shape that generate, for example, a disk of the size of a single cell, but also multiple disks with the idea of activating multiple cells which are close to each other. So laterally, we can be very precise, but now if you look at the axial resolution, you will see that in the first case, we do have a relatively good axial resolution, while here, the axial resolution is largely deteriorated. And now when this same beam goes through a grating for temporal focusing, we can nicely concentrate the absorption around the focal plane. And so that independently on the lateral spot shape, we can get an axial resolution which is compatible with the size of a single cell soma. Okay, so this is to control the activation of multiple cells in a plane. And now that, as I was saying before, we would like also to reach multiple targets eventually in 3D. So the system requires another modification, which is the addition of a second spatial layer modulator. So this device alone will produce a distribution of multiple diffraction in the spot, each of them centered at the location of the target that you want to activate. And now you could here add a galvanometric system and perform what I was saying before, this multi-spiral scanning approach, or we can put this special layer modulator after a first one that created, in the way I was showing before, a certain shape on the grating for temporal focusing. This shape is going to be temporally focused. And so in this case, what the second special layer modulator does is to multiplex all what is created at the temporal focusing grating at the location where we have generated this multiple point. And so in this way, it is indeed possible to generate, and this is just an example, 50 temporally focused disks in 50 different axial positions. So to be mentioned, there are multiple variants of this schema I'm showing here, where the difference among them is mainly in the way this first part is done. So this can be an expanded Gaussian beam. So we speak about 3D shot, or can be more complex phase modulation approaches. And I won't have time to compare them. You can see that the comparison in this review, but in general, with each approach with its advantage or limitation, they all give the capability today to target hundreds to generate hundreds of axially confined spots in millimeter cubic volume. So from the photonics point of view, we are on the way to control in a very precise way light patterning in 3D in large region at multiple spots. So now to combine this approach to have a true single cell resolution optogenetics, this required to integrate the progress made in the light delivering approach with the progress made by many labs that in the past year have worked to find strategy to develop what we today call soma-restricted opsin. So these are strategies that enables to confine the expression of the opsin selectively into the soma, so that when we can combine this with a precise light shaping, we are sure that only the illuminated target get activated, while in the other case, we could also have processes from the surrounding cells that are crossing the illumination volume with obviously a drastic reduction in cellular resolution. So here there is an example. Here we are using a soma-targeted opsin produced by the lab of Ofer Izar, but this is just an example, visual cortex in vivo, and you see that, for example, here we can choose those 3 cells while recording their calcium response, and then just select one in between the 3 cells, and only that one will keep on responding. So we are really on the way to combine light shaping and molecular focusing to get the control of neuronal activity with a cellular resolution. So the application of this approach, we are starting to see more and more examples where this kind of approach are used. So there is a first number of applications where the single cell resolution was a key importance, and this was to perform high throughput connectivity mapping. There are examples where this was done in the retina, in the zebrafish larvae, in mice cortical slices. And so in this case, the idea was to use holographic single cell optogenetics to activate a specific sequentially one of multiple presynaptic cells while recording either using electrophysiology or functional imaging the response evoked in the postsynaptic cells. there is another large class of experiment that take advantage of holographic light multiplexing, where this approach was used to probe the threshold of perception. So there have been fantastic papers coming out in the past year where this was used again in different brain regions, visual cortex, olfactory bulb, or hippocampus. And in the first case, this kind of experiment were done by activating 1 to a maximum of 10 cells at a time. In the second class of experiments, this required to increase the number of achievable targets, so that typically the number of cells activated were between 2 to hundreds of cells. So, okay, this is more or less where we are today with circuit optogenetics. The technology is pretty robust, and it starts to really be used in answering key biological questions. Now, in the remaining part of the talk, I would like to discuss 2 main points about this kind of technology, and this is how many spots we can realistically reach. So how big are the ensembles of neuronal ensembles that we can manipulate? And the second is how quick we can perform sequential light patterning. So with the idea is can we actually use this approach also to start seeing the importance of the synchronicity in the spiking activity produced in the case where we activate multiple cells. So with respect to how many spots, the question we already shown a few years ago that a big improvement was done when using a low repetition laser, because those laser enables, given with the same average power, to increase the extension peak power. And so these already give a big improvement in the number of achievable targets. Also because those lasers can today deliver a lot of power. So we can have typically about 25 microjoule at the objective exit. And so if we consider that typically more or less for any value very similar, and depending on the opsin used, we can activate neurons with about 20 nanojoule per cell. So if you do the calculation, this gives you theoretically the capability to activate 500 to 2,000 of cells. Okay, so the reason why I'm saying that this is a theoretical number, because we have to take into account 2 important factors now. One is the scattering. So obviously this number that I'm giving here, it doesn't take into account the scattering, but now if you want to reach a target which is a 1-2 scattering length deep in the brain, this number has to be multiplied by a factor of 10 or even 20. So this will obviously drastically decrease the number of achievable cells. And the second important point is the thermal damage. So at a certain point, if what we have in mind is to manipulate neuron ensembles of hundreds of cells, we have to take care of not exceeding the single Kelvin thermal photodamage threshold. So we try to work with these 2 limitations, and eventually to propose solution to overcome these 2 limitations, we recently developed a variant of computer-generated holography, which we called FLIT, fast sequential light patterning. So here the system is very similar to what I've shown before. So we have a multiplexed spatial layer modulator as before. We have a grading for temporal focusing. But there are 2 key differences with respect to the system I've shown before. One is that now the multiplexing spatial layer modulator, instead of being addressed with just using a single hologram, is addressed with a number of vertically aligned holograms. We tested the configuration using up to 25 independent holograms. And the second variant is that now we have a galvanometric system that enables to scan the temporary focus line across these 25 or multiple holograms. And so in this way, because we can quickly scan the temporary focus line across those multiple holograms, we can generate faster sequential light patterning with a temporal resolution which is as short as 90-100 microseconds. Okay, so now why this should help in increasing the number of cells and minimize thermal damage? This is something you can understand once I have explained you this the idea of cyclic illumination. Okay, so this is something that we recently demonstrated, and the idea of cyclic illumination is the following. Starting by what is done in conventional two-photon optogenetics, we can say that in this case, neuronal activity is typically controlled by using an illumination pulse of 5 to 20 milliseconds, okay? And now the idea of cyclic illumination is to, instead of having a continuous illumination, is to illuminate the cells with a series of short, let's say about 50 microsecond pulses. And it can be shown that if in a conventional to photon optogenetics, we need a certain power to activate a single cell or a single group of cells. In cyclic illumination, we can produce exactly the same photocurrent if each pulse is delivered with a power which is equal to the power of conventional to photon optogenetics by the inverse of the duty cycle, where the duty cycle is defined as the ratio between the time between two pulses and the duration of a single pulse. Okay, so why this is an advantage? Because if we can do so, we can actually produce the same photocurrent, but now we have a certain time between one pulse to another, so that with the same power that we used to illuminate the first hologram, we can now go through the different holograms addressing the spatial A modulator, and then going back to the first hologram. And so by doing so, in the same time that we would excite a single cell or a single group of cells in conventional to photon optogenetics, we can now excite H more cells or group of cells using cyclic illumination. So the first step was to demonstrate this key factor. So can we really increase reduce the necessary power of a factor of a square root of H? So we performed multi-target photostimulation with calcium imaging, and indeed we showed that the power needed in conventional holography is a square root of H times higher than what needed in FLIT, and we did this for multiple holograms. And so this has two important implications. Okay, so the fact that in conventional holography, if you want to excite H cells or H group of cells, we should multiply the total power by this same factor. While in cyclic illumination, if we want to reach the same number of cells, we can only use square root of H more power. And so this has two implications. The first, that we can obviously reach square root of H more cells. And the second is that even if we want to reach the same number of cells, we can reduce of square root of H the total power sent into the sample, and so minimize the thermal effect. Okay, so the system we have worked till now, we tested the system, and we showed that we can, for the present configuration, address up to 25 independent holograms without having any deterioration in the spot shape or axis resolution. So this means that we can, in principle, reach at least 5 more cells, and I will discuss about the thermal damage afterwards. So this factor of cells becomes to be pretty important if you look at what happens with the depth. So imagine that you really want to control a large neuron ensemble. So if you want to control the 5,000 cells that are possible today with existing technology, you have to consider that if those cells are distributed in a large volume, many of them will be very deep. And so for them, the scattering will require much more power. And so this number that seems to be pretty high actually will go quickly down to less than 100 cells if your cells are distributed in a large volume. So this factor of 5 or more that we can gain with cyclic illumination is already enough to bring back that achievable number of cells to a few hundreds of targets. And now the second point I would like to underline is the effect on the temperature. So this is something that becomes to be important when you really want to control large neuronal ensembles. And this is something we are already started to investigate a few years ago, where we developed a theoretical model to evaluate the thermal rise into photon optogenetics. And we validated this model experimentally, first in vitro and more recently in vivo. And so we showed that in the case of single cell photostimulation, these are the continuous line are what is predicted by the model, and the dotted lines are the experimental data. You will see that actually is the temperature rise characterized by single cell excitation do not exceed the single Kelvin, knowing that typically the condition of a typical to photon experiment are more in between the green and the blue lines. Okay, but then what happens when we start to increase the number of cells that we want to control? For example, if you go to up to 100 cells distributed in a volume typically accessible with computer-generated holography, you will see two effects. The first one, that the absolute value is much higher. So now we can easily reach up to 10 Kelvin. But the second most important effect is that the decay of the temperature rise is now much longer than the case of an isolated spot. And now, because the thermal damage not only depends on the absolute temperature value, but also on the illumination time, this increasing of the decay times also can have dramatic effect on the produced thermal damage. So now the use of cyclic illumination have two advantages with respect to this huge increase of temperature. The first one is that at the level of a single cell, because the illumination is divided with the short pulse, so we have a more efficient thermal dissipation than in a conventional two-photon optogenetics. And most important is that the fact that because of reaching the same number of cells require square root h less power, this allows to strongly decrease the total temperature rise. So these are the simulation for a different number of holograms, and in greens is the simulation for these 25 holograms that we experimentally demonstrated. And so you see we can drastically reduce the total temperature rise. Okay, so this is as far as it concerns the use of FLIT with cyclic illumination to increase the number of targets and reduce the thermal damages. And in the last few minutes, I would like to discuss another configuration that can be combined with together to this FLIT approach. And in this case, this was to solve or to increase the resolution for sequential light patterning. So as Raphael was saying at the very beginning, in the definition of a neuronal ensemble, so one way to define a neuronal ensemble is a population of cells that fire together, okay? So now what would be interesting at a certain point is to see what does it mean together? How long is this together? So how closely those cells need to fire to be considered an ensemble, to have the effect of a neuronal ensemble. And so at this point, we are at the limit of the temporal resolution that we can technically achieve. And this is because there is a first limitation given by the refreshing rate of the spatial layer modulator, but most important is the limitation given by the illumination time. So we can, for what I was showing before, using this galvanometric mirror, create a number of patterns that can very quickly refresh with a temporal resolution of 100 microseconds. But unfortunately, this is not enough. So even if we would have an ultra-fast spatial layer modulator, still the limit in the temporal resolution for sequential light patterning will be the illumination time. And so for that, what we propose is to use this approach together with what we call the hybrid illumination. So here the idea is the following. I will show this for the case of two cells that you can think is the same if we were speaking about two groups of cells, and then we will see this can be generalized to a larger number of cells. So the idea is the following. Imagine that you want to precisely control the relatively spiking time between cell number 1 and cell number 2. And imagine that those cells will require 5 to 10 milliseconds to be activated. So this will be your limited temporal resolution. But now we can do the following. We can use a first hologram that is illuminating cell number 1, a second hologram is illuminating cell number 2, and an intermediate hologram which is illuminating both cells. And now we imagine that we need the cell number 1 to spike. We need to illuminate cell number 1, let's say for 5 milliseconds to have a spike, and the same cell number 2. What we can do is to now switch our illumination beam through these different 3 holograms so that the total time spent on F1 plus F2 is this 5 milliseconds, and the same, the total time spent in F2 plus F3 is this 5 milliseconds, so both cells will be able to spike. But now we can stay on the first hologram at time as short as the 50 microseconds given by our galvanometric system. And so this now is the minimum time you can introduce in between the two spiking time. And so we basically can desynchronize the spiking activity with a temporal resolution, which is now only limited by the illumination time of the first hologram. So we validate this approach in electrophysiology first. So this required to patch 2 cells belonging to those 2 groups of cells illuminated by the hologram F1 and F2. And actually we show that we can reduce the illumination time of the first hologram down to these 100 microseconds and be able to precisely detune the relatively spiking time of the 2 cells. So this for obvious practical reason, we could only probe the spiking activity among 2 cells, but the principle can be generalized in multiple holograms, so that actually can be shown that we can control the relatively spiking activity among n cells or n group of cells by using 2n minus 1 hologram. So technically today, this corresponds to about 13 group of cells or of cells. So this approach is really open the way to perform a very precise control of the relatively spiking time. And there is another interest of this approach is that if we really go with a cyclic illumination towards a situation where we now want to pattern 500 of cells or even more, to don't be again in the first limitation that of wide field illumination where all the cells are synchronously activated, this approach could also simply be used to introduce an arbitrary desynchronization between these multiple cells. Okay, I think it's time to conclude now. So I would like to conclude just by saying first the conclusion. So the first general conclusion that really in the past year, there has been joint progress in many disciplines, away from shaping, but also laser development and ops in engineering. And these three disciplines together gave us today the possibility to really start to manipulate large neuronal ensembles in 3D with cellular precision and millisecond temporal resolution. And I have shown you that for multi-target, multi-cell targeting, we can increase even more the size of the ensemble that we can manipulate by using this fleet approach together with cyclic illumination. And then if we want to start to play with the time code, we have proposed recently this hybrid illumination that enables to very precise control the 2 millisecond precision, the tuning of neuronal activity. Of course, there will be still further direction we are going on to increase even more the field of view, to go deeper by performing holographic endoscopy. I only speak about activation, but it is also now becoming more and more important to be able also to inhibit or even to do at the same time inhibition and activation. And then eventually to monitor what we are doing in a very precise way would be also important to develop approach for fast multi-target functional imaging. So I would like now to thank the people of OMLab, specifically here in red are the people implicated in the new data. So they have been, I would like to thank for this Emiliano, Cécile, Dimitri, and Clément and Christophe for building up and optimizing and characterizing the fleet microscope together with Giulia, Massilia, and Cécile again to validate the use of this approach for both cyclic illumination and hybrid illumination, and Benoit Forget for developing all the temperature modeling. I would like also to thank obviously the financing source, and thank you for your attention.
Thank you, Valentina. Let me start you with a question. In terms of, are we limited by the instrumentation? Imagine that you were running the US Brain Initiative and you had a budget, I think the current budget is $620 million for this year. What would you do with SLMs and with lasers?
Yeah, good question. So with lasers, I think that okay, there will be always need of more power if we want to increase the number of cells, or even the depth at which we want to reach targets. So I will go on with the improvement of deliverable power. What is missing is probably also powerful laser, because most of these fiber lasers are in the 1030, 1060 nanometer. And what is missing is a laser delivering a comparable power more in the range 920 nanometer. And this will be interesting because we could will be easier to, for example, use blue shifted optics with red indicators. What I would do with respect to the SLM, I think there would be nice to develop larger SLM. Because this is the limiting factor with respect to the achievable field of view. So I think for sure the larger is the SLM, the more experiment we could do. So for example, in the case of our fleet, we were limited about this 25 hologram, while larger SLM would enable to increase the number of holograms, so increase the number of cells. What else would I do? So from the technology, you were saying lasers and SLM, then I think that also if we can call this technology, I think what would be really nice to improve all what is the homogeneity of the expression. I don't know if this is true transgenic animals or to more efficient procedure to for viral expression. But this for sure, I mean, because at a certain point now we can generate multiple spots. We can nicely control the precision, the axis resolution, no matter which approach we use. But at a certain point, at least in our end, the limiting factor is really the dishomogeneity on the expression. So it's very difficult to control what is really happening. And so when you want to stimulate the 500s of cells, you can't characterize the latency and the jittering for each of your 500 elements. So you could only rely on a good homogeneity of the expression.
How about the optics, the objectives?
Yeah, the objective, that's right. This is another point. So objective with a large back aperture are always good, with a long working distance as well, so that they are also compatible to eventually electrophysiology or to reach deeper brain area. So there is also some progress to be done on the objective development. That's true. And then I didn't really develop this much, but in respect to if you want to reach deep brain region, of course the solution is triphoton, but still will be limited on the first millimeter. Then there is all the field of endoscopy. And again, there is a compromise between field of view and numerical aperture. So I think also on green lens, there will be probably some development to be done. So to increase the field of view without the cost in numerical aperture.
I wrote everything down and I will send it an email to the director of the Brain Initiative, which never answers my email. So don't get your hopes up. So any more questions for Valentina? Unfortunately, we're running a little bit late. Emre, you have a question?
Yeah, just quickly, the sequences are incredibly interesting, but one could imagine that you have these almost unlimited possibilities of combinations, not just permutations, but also the orders of these things. So you must have thought about these things, how fast or how many of such sequences one could test using these methods, and how long the circuit could last without getting laser damage?
Sorry, I missed the end of your question.
I imagine that at some point laser damage will start building in, and we are looking at these. I imagine that there must be some smart strategies to go through multiples of sequences, because they can get unlimited, right? If you're dealing with 100 cells and their orders. Are you also thinking about strategies of how one can assess different sequences that actually may have different properties as you're sort of sequentially activating these networks?
So your question is, what is going to limit the number of sequence that you can test, because at a certain point there will be some damage of the sample? This is what your question?
Yes, and because it can get too many sequences, like very, very sort of drastically. I guess there must be some smart strategies to go over different sequences in a smart way.
So,
have you?
Yeah, with respect to what you say, the damage, you said something like the damage will keep on building up. I'm not so sure if your configuration is well designed. So the nonlinear photo damage, I think we are pretty safe. This is something my lab and others are carefully characterized. I think there is still a quite large margin to be below the nonlinear photo damage threshold. The temperature, I just showed you that what is really important is something that can be nicely controlled. If you take care that the relative distance of the target is larger than the thermal diffusion length, so then the fact of stimulating 500 or thousands of neurons doesn't really make a huge difference. And because the thermal dissipation is quite quick, I don't think it's an effect that will build up during the experiment. Okay. So now, with respect to the sequence, yes, there are now. I mean, the number of experiments you can do with this approach are very large. So I don't have an answer to all the possible configuration you can think. But for example, if you want this approach to probe connectivity, for example, there are clever ways to go from one cell to another, or from one group of cells to another, so that the frequency at which each target is excited is much lower than what would modify the structure of the network. But indeed, and this is probably also a way to answer to Raphael, what is missing from the technological point of view, all what I'm saying now is with respect to this photostimulation approach, but we have to think that in most of these experiments, we will perform a sort of all optical. So it's not only what the photostimulation laser is doing, there is also the imaging laser, there is the crosstalk. So what is the imaging laser is doing to the network is also extremely important. So this is something that has to be minimized. And so, yeah, I mean, we have to consider that there is a relatively new technology, and there are many things that have to be carefully considered. So for sure, what you say is difficult to give you a general answer, you know? But indeed, if we start now to think of manipulating large ensemble for long manipulation section, this has to be done in a very careful way. Yeah.
Thank you, Valentina. We'll have to now move on. So you could argue, someone could argue that just as important as experimental methods are theoretical and statistical and mathematical methods to look at ensembles. And the next speaker has pioneered very beautiful methods. I guess one common thread of this first session is beauty in terms of the data, the elegance of the approach. And now we're going to be listening to Carsten Stringer, who comes to us from Janelia, and she will discuss analytical methods to visualizing the geometry of neural activity. Carsten, you have 25 minutes and then there will be a Q&A. Thank you.
Sounds great, thanks. Maybe set my timer. Okay, hi everyone. Nice to see everyone here today. I'm a group leader at HHMI Janelia Research Campus. I'm gonna be telling you about work done in collaboration with Marius Pakisariou, Lin Zong, and Attica Saida. And so sort of going with the theme of the workshop, finding neural ensembles or visualizing the geometry of neural activity will hopefully give us insights into the sorts of computations these brain areas are doing. And so this visualization problem is important because we've entered this era of big data and neuroscience. So we can record simultaneously over 100,000 neurons in the zebrafish. We can record tens of thousands of neurons in the mouse brain. These are both with calcium imaging techniques. We can also get single cell RNA sequencing information from thousands of neurons. We can find structural information or connectivity information in the fly, for instance, and we can record high dimensional video data from animals as we're doing these large scale recordings. And so what are we going to do with all of this data? So perhaps deep learning can help us. So in fact, the rise of deep learning has coincided with this era of big data and neuroscience. So deep learning is good at solving an engineering problem, like is this picture a cat or a dog? Or a segmentation problem, for instance. So this is what we do in our algorithm CellPose for people who need to segment cells. We have an algorithm that uses deep learning to solve this problem of finding outlines for cells, or you want to track key points, for instance, on a mouse's face. These are all kind of engineering problems where you know what the solution should be. It should be, you should correctly, for instance, track these points. And you have specific labels that you give the algorithm to learn. And so these are all supervised problems. Keep in mind though, even with a supervised problem, the neural networks are only as smart as the training data that you give it. So they still will get confused. And further, we don't have an engineering problem to solve here in interpreting this large scale data. We actually have an unsupervised learning problem where we need to find patterns that a scientist can interpret and gain insight from. And we don't know what these patterns are going to be a priori. We don't have cat or dog labels on our neural ensembles. And so the most common, so we're going to try to come up with a good unsupervised algorithm for this large scale data. So I'll start by showing kind of the most common approach for unsupervised dimensionality reduction for visualizing data, which is principal components analysis. So PCA reduces the dimensionality of the data to the dimensions which have the most variance. So we're going to look at PCA applied to some neuroscience data from the Hantman and Branson labs at Janelia. And so they collected neural activity while the mouse was doing this reaching task. And they took the top principal components of the neural activity, and they also looked at where the hand position was. of the mouse. And then they could look at how these principal components varied across, for instance, different optogenetic simulations to see how the neural activity varied. And so we can just stop this movie. You can see, so the PC1 is this direction in the neural activity, which has the most variance, that the neurons vary the most across. This next dimension that PCA finds is the dimension with the second most amount of variance, and so on. And so you kind of, this plot is sort of, has 3 dimensions, and what we're missing on this plot is actually the PCs above PC3. And so we may be missing these high dimensional dimensions by just taking the top 3 PCs and plotting them. And so in some cases, maybe we're not actually missing them. So for instance, if the neural data has a principal components, has an eigenspectrum, or the variance of these PCs follows this power law, or sorry, follows, basically goes to 0 after a few PCs. So you see the variance is high here and then goes to 0. Then we can basically compress the neural data into a few dimensions and visualize those dimensions, and we'll get all of the information out of the neural data. But that's not actually the case with real neural data. So we take the principal components of this large scale 10,000 neuron data in response to thousands of natural images. What we find is that this eigenspectrum follows a power law decay. It doesn't just go to 0 at some point. There's significant variance in these higher dimensional PCs, and there's high dimensional activity in this circuit that we're losing if we just visualize the top few principal components. And this isn't just a feature of stimulus-driven activity in mouse visual cortex in response to natural images. We found this power law decay of the eigenspectrum in a variety of recording settings. So for instance, in spontaneous activity, where the mouse is just sitting on the rig and we're recording neural activity, we also see a power law decay. If we look at zebrafish whole brain activity, we also see a power law decay. In the single cell RNA sequencing data, where you have a matrix of neurons by gene expression, you have a power law decay. If you look at the mouse behavioral video, you have a power law decay, which is inherited by the fact that natural images also have this power law decay. If you look at correlations between words and sentences in the English language, you have power law decay, and also in social networks, in the edges of friends in a social network. And so what are we going to do with this high dimensional data? We can't just use PCA. What are we going to do? And so Jeff Hinton actually had a joke where he said, to deal with a 14-dimensional space, we'll just visualize a 3D space and say 14 to yourself very loudly. Everyone does it. And I mean, this is kind of a tradition that lots of people follow. And actually, this was also kind of a funny tweet that this person found, was actually this tweet, because they called him the godfather of AI in this tweet, it was actually classified as a crime film. And so be a little wary of deep learning in general. And also, I thought it was kind of another meta joke, is that this is a crime against data to just take a 3D space. But anyway, okay, I'll stop with the bad jokes. What are we going to do with this large scale data? Okay, so maybe we can use other unsupervised approaches. So for instance, we can use nonlinear dimensionality reduction, and some of you might be familiar with t-SNE, for instance. And so we could take, for instance, this matrix of neurons by genes and smush it into a 2-dimensional space where we put neurons close to each other in this space that have similar gene expression patterns. And this is done in a nonlinear way to try to preserve as much as possible the high dimensional structure of the data. And so this is what this looks like for this gene data from the Allen Institute. You can alternatively use a clustering algorithm, which runs directly on this matrix of neurons by genes, to try to find clusters of neurons which have similar gene expression. And so you can actually take the clustering output and color each neuron by which cluster it's in on this plot here, and that's what's being shown. And what you can see is that t-SNE isn't perfect in this case. So the high dimensional, these clusters that were found in the high dimensional data aren't always preserved perfectly, and all those neurons aren't always put close to each other. You can see kind of, particularly in this area, there's kind of speckles of neurons that maybe are not quite perfectly put next to their neighbors in the high dimensional space. And so we kind of, so what's the right approach here? We thought maybe we should try to do both of these in order to preserve high dimensional structure, but also come up with a way to actually visualize this data. And so our method raster map, the first thing it does is it clusters the data to find these clusters in high dimensional neural space. So this is a raster plot of neurons by time. And you can see the running speed is put here up here. This is spontaneous activity, by the way. So this is mouse visual cortex activity while the mouse is running, can run, whisk, sniff about it, but it's in total darkness. We're not showing visual images. And the first thing we can do is we can cluster the data. And so this is what the clusters look like of neural activity. And then what we can do is we can sort these clusters so that clusters with similar activity are put next to each other, like solving that T-SNE problem. And so when we do that, we get a picture that looks like this, where we have across this, you can see neurons that are active, for instance, at the beginning of running that are put next to each other. This is thousands of neurons. You can see this scale bar is 5,000. So we're capturing this global structure, but we're also putting neurons close to each other that locally have similar activity patterns, like maybe even this small cluster here, which is kept together, that has specific activity. And so we'll get into kind of a little more detail about in a second about what these patterns are related to. Spoiler alert, it's behavior, but let me go through how the algorithm works. All right, so the first step is, as we were describing, you cluster the data, and we found that k-means clustering works the best with a cosine similarity distance. And so we get 100 to 150 clusters from these 50,000 neurons. And then the next thing we do is we rearrange these clusters so that clusters with similar activity are put next to each other. And so we can get this cluster by cluster correlation matrix. So you can imagine, so we have like 100 clusters, we have a bunch of neurons in each cluster. And so we have an activity trace for each cluster, which is the average of the neurons in that cluster, like a neural ensemble in a sense. And we get the correlation between these clusters. And that gives us this matrix here. And now we're going to sort this matrix in a particular way, actually, not just to put clusters that are correlated to each other next to each other. We're also going to try to preserve global distances. And this is actually a matrix that will do that, which has a power law decay of the PC of its PC variances. So it has an eigenspectrum like the neural data, which has this slow decay off the diagonal. So it's not just going to put the most clustered things next to each other, just preserving the diagonal. It's also going to preserve off diagonal correlations. And so you get a matrix after the sorting that looks kind of blocky like this, where you can see kind of it decays off the diagonal. It's not just red right along the diagonal. There's also these global patterns that are preserved. And then, so this clustering algorithm only works, well, it works with as many clusters as you want, but it will start to get slower. So with 150 clusters, it takes less than a minute. As you get to 200, it'll take 5 minutes and so on. So it's an iterative optimization algorithm that for an NP hard problem that will start to get slower. So to kind of get around this problem that we're limited by number of clusters, we upsample these cluster centers. And so so each cluster center, we can we have them in order, and we can do a linear interpolation between one cluster center to the next cluster center. So this is what the upsampling looks like in the PC space. And then we can assign each neuron to one of these positions, whichever one it's most correlated to along this space. So if we upsample by a factor of 10 and ran with 100 clusters, then we'll have 1,000 positions to assign neurons to. And so ultimately we get a picture that looks like this, that hopefully will help us gain insight into our data, and didn't make any assumptions about the structure of our data by performing this algorithm. Okay, so how can we tell if this algorithm works well? So we can use benchmarking techniques that are commonly used in dimensionality reduction techniques, such as these neighbor metrics. And so we have, we can look at this toy data, which is 3-dimensional, where every neuron has a response, in a sense, to these 3 different stimuli. And you can see that we've got clusters of neurons with similar responses, which are color-coded in these different colors. And so if we want to preserve local distances, like we've been talking about, like these high-dimensional clusters doing a good job with that, then we want to make sure all the blue points are put next to each other, the green points, the yellow points, and the red points. And so we can quantify how well the algorithm does at putting neurons close to their neighbors by asking how many of my original neighbors in this high-dimensional space are still my neighbors in the low-dimensional space. So we can kind of take this 1 point here, see what its closest neighbors are in the high D space and see if they're the same. And we see what fraction are still their neighbors in the low dimensional space. And we find that raster map outperforms t-SNE, UMAP, and PCA in preserving these local neighbors. And so we don't just care about preserving local neighbors. We also care about getting a good global picture of the space. So we might want to zoom out and look at hundreds of neighbors rather than the top 10 nearest neighbors. And what you can see maybe from this this plot already. So we have blue points next to green points next to yellow points. So it looks like we're preserving some of these global distances here. And so we can check if that's true in the actual data, not just our toy data. And oops, sorry. And what you can see is that raster map does just as well as or better than t-SNE, UMAP, and PCA at preserving these intermediate and global neighbors, so the 100 nearest neighbors or the 1,000 nearest neighbors. And so you can run t-SNE with different perplexities, and we also tried that, and with running multiple perplexities at the same time, and we still couldn't get an algorithm that outperformed raster map. There's always kind of this trade-off between the local and the intermediate score. And so we found that raster map was the best algorithm for neural data, we think. Okay, and so I'm just going to show some examples of applying raster map to various data. And so the first one I'm going to show is for visualizing behavior predictions. So I've been showing you that neural data of mouse visual cortex in the dark. And we basically, while the mouse is in the dark, we're recording its face. And so we can track the key points on the mouse's face. And we can take these key points and fit these key point movements basically with this model to best predict neural activity. And so we fit a model, we use 1D convolutional filters to fit the various movements of these key points. And we ultimately get a model that basically works better than our previous approaches. We've used in the past, for instance, the principal components of the face, but we found that this model with these key points actually works better. And we can now visualize, it helps to visualize these predictions with raster map to see what we got right and what we got wrong. And so this is what the raw data looks like, visualized with raster map. And then this is what the predicted data looks like. And so we can see that which features we're capturing well. Maybe we can zoom in and look at things that aren't captured well. We can see what timescales things are captured at. This allows that kind of visualization that may not have been so easy to do in the past. And again, it also allows you to say, what does this number of 40% variance explained mean? It's a lot easier to kind of see it qualitatively what it means in a picture. And so another thing we've applied raster map to is whole brain data, for instance, from zebrafish data collected in Misha Aaron's lab. We can sort neurons here across the whole brain. And this is an experiment where the fish was shown a lot of different visual stimuli. And there's periods where the fish is also swimming in response to these visual stimuli. So these are kind of the tail neurons. And we can color, because we have this one-dimensional sorting, there's an advantage here that we can color neurons based on their positions along this sorting. And so you have this raster map position sorting here. We can color these neurons in this fish brain based on their positions. And this can start to give us an idea of what kind of maybe which parts and clusters in this raster map correspond to clustered areas in the brain physically, or maybe correspond to patterns that drive neurons across the whole brain. We can also apply this approach to wide field imaging data. So it doesn't just need to be applied to single neuron data. We can take voxels from wide field imaging and treat them the same way that we've been treating single neurons. And we can get a sorting that looks like this. And again, we can color the voxels here by their positions on the raster map. And so this is data from Anne Churchland's lab where the mice are performing an auditory discrimination task. They're also being shown visual stimuli as well during this task. And so you can see lots of different neural patterns. Some are more obvious than others. This one, for instance, looks like a visually driven pattern. This is this purple part, which corresponds to primary visual cortex. And so doing a sorting like this allows us to kind of give us a parcellation of the brain that's sort of independent of brain areas from some sort of atlas. For instance, we can say these are groups of voxels that have similar activity, and kind of maybe look at them at that level. And it also gives us a figure that we can look at that maybe it's a little easier to interpret. All right, and so this is all for data that maybe does or doesn't have sequences. We also made an extension of this algorithm that works well for data with temporal relationships, or in particular, maybe sequences of neural activity. And so all of the correlations we were talking about in the past between these cluster activities were always instantaneous correlations. So at a time lag of 0. So were those clusters correlated to each other in instantaneous time? But we can also compute the cross correlation of clusters. So take different time lags of these cluster activities and compute the correlations between these cluster activity traces. And so that can give us a plot that looks like this, which maybe, for instance, has a peak that's not necessarily at 0 at the instantaneous correlation. And so we can take we can take the correlations at various time lags and take the maximum correlation over these time lags for each of these correlations between cluster centers. And then we're going to, again, sort this matrix after taking the max over time. And we're going to add an additional penalty here. So we want this matrix to preserve global information, but we also have this kind of traveling salesman additional kind of penalty we add, which says that you should also be very correlated with your nearby clusters. And this is kind of like, you can change this penalty in the algorithm. It's kind of like the perplexity parameter in t-SNE. So you can try to make things more better preserving local structure rather than global structure. And so we can apply this algorithm to data where there are in fact sequences. So this is neural activity from recorded by Lin Zhang in Marius Pagliussari's lab, where the mouse is running through a virtual corridor, in this case with leaves textures on it. There are 4 different corridors in this task. There's a leaves texture and a circles texture that the mice have seen many times. And then there's these 2 new textures, which are new exemplars of the leaves and the rocks that the mice have never seen before. And so there's 4 corridors in total that the mouse is running through. And we can kind of see the sorting of these clusters in this plot. Let me show the upsampled version of this plot. I think it's easier to see, and now it's colored by the different clusters. And so there's the blue corridor and the red corridor are the 2 corridors I was saying the mouse has seen before, and then the teal corridor and the purple corridor are the corridors that the mouse is seeing for the first time. And what you might see is that there are these sequences that rastermap was able to find in the data. There's breaks in these sequences actually when the mouse is getting a reward in this corridor, and that the mouse actually stops to collect the reward, and then the sequence continues. And so you can see these 4 sequences that rastermap found that can start to give us insight into the sorts of visual coding that's going on in the brain. And there's also other interesting different signals that Lin and Marius found in this data using rastermap and that they hadn't seen before. For instance, they saw neurons that were active before the mouse collects the reward, but then immediately turn off after the mouse collects the reward. So you can see these neurons here. They're also active in this new corridor that the mouse has never seen before, suggesting that the mouse thinks that this new corridor is also rewarding. I'm not going to go into that in more detail, but basically they were able to find signals that they weren't expecting to find, but were able to find with rastermap. And so this is kind of a good way to visualize your data to find signals that you maybe weren't necessarily expecting to see. You can also apply this, so the sequence finding, to data artificial brains. So here's an example of an artificial neural network, a QRDQN network playing Space Invaders. And so this network has a convolutional neural network and then also a value network. And these sequences are happening in the convolutional neural network in these are basically feed-forward sequences that are generated by the correlations in the visual space that the agent is processing. And so that's similar, we think, to the visual sequences that we see in the mouse brain, that they're kind of more feed-forward driven sequences. You can also see these, another agent, you can again see these sequences. This one's playing sequest. You can see the value network kind of going down as well, maybe reminiscent of value signals in the mouse brain. I'm almost out of time, so I'm just going to quickly explain this and show the data and then wrap up.
Yep, sounds good.
So basically what I've been showing you with the spontaneous activity is we can look at where these neurons are in their first 3 PCs, which again, I said didn't capture all the information, but maybe it'll help us visualize the raster map. And what you can see is that, so each of these dots is a neuron, and you can see kind of the trace that raster map takes through this PC space. So we start at white and we kind of travel around. And so you can think that raster map is sort of making, we have this kind of ball of activity or of neurons here in this PC space, and raster map is kind of finding like a space filling curve almost, it looks like, through this PC space. And so here's an example of a space filling curve. And so we thought to try to make raster map work for even higher dimensional data, we wanted to basically work with more clusters, maybe thousands of clusters. And so we run raster map iteratively. So we run it with 100 clusters initially, and then we divide it into 4 parts, and then we run raster map again on those 4 parts, and then we can divide again and again, and so on, and get more clusters and get higher dimensional data that we can visualize. And so I'll skip over the kind of toy data because that's less exciting, but basically t-SNE failed on this kind of toy data. I'll just show the stimulus, the actual neural activity. So this is neurons by time, and there's stimuli being shown, thousands of natural images being shown to the mice. They're also free to run. You can see the running speed here. What we found was on this data in particular that raster map really outperforms t-SNE, especially on the local score. And you can see the neurons, there are similar neurons put together based on running correlation. This is their correlation with running speed. This is how reliable these neurons are in response to visual stimuli. This is how well they're fit by a linear model from stimuli to neurons. And then we can just kind of look at the receptive fields in this space, and you can see kind of smaller receptive fields put down here by raster map, bigger receptive fields up here. And we can kind of zoom in and even see, like, as you're going through the raster map, you see kind of the rotation of these receptive fields in the raster map. And we can again visualize kind of the raster map position on the brain and see that you have neurons in primary visual cortex are the ones with the smaller receptive fields, and the ones up here are the neurons with the bigger receptive fields. And we also have a GUI that you can use to run raster map. And in summary, we found that raster map quantitatively captures local structure better than t-SNE and UMAP. We have extensions of the core algorithm for finding sequences and find structure in neural data. And we found these novel and interpretable cognitive neural signals that hadn't been seen before. And so I'd like to thank my collaborators, Marius, Lynn, and Attica, and HHMI Janelia for my support. And thanks everyone for listening. Sorry, I'm a little over time.
No, you're perfect, Carson. We're the ones who started late. So there's a question from the audience, and anonymous attendee. When you refer to clustering the activity of the neurons for calcium imaging, what have you found is the best form of data to cluster? The raw data, the raw fluorescence, delta F over F, peaks of activity, etc.
That's a really good question. So what we found is that the deconvolved data works the best. In general, what we found is that if you use the deconvolved data, so for instance, when we look at this stimulus SNR, how reliable neuron responses are, we find that the deconvolved data is more reliable than using the raw data. And so we think it's a better form of the neural signal to use if you've got decently high quality data and clear transients.
Just to make sure people understand what you mean by deconvolved data.
Oh yeah, sorry. Yeah, so when we're recording neurons with calcium imaging, there's a long decay time of the sensor. And so the signal goes up and then it decays very slowly. And so you can get rid of this decay with deconvolution, which basically will then, if you just had this spike here, it will kind of find those spikes in the data and get rid of those kind of longer tails. And so that can help to in general, also the pictures look nicer, but it also can increase your SNR because then you're not getting this bleed through of these long decays into other stimuli, particularly presenting stimuli quickly. So it's an important step to do, I think.
By the way, Carson, your data looks beautiful, I have to tell you, including these pictures. I think there's a question from Emery. Emery, you want to go ahead? Yeah.
Carson, fantastic stuff. So when you identify these ensembles of sequences, do you also identify where they start and where they end? And if so, how are they distributed? Like what kind of time domains of sequences can you identify? Are there like binary distributions or like very smooth in terms of length of such sequential ensembles?
Yeah, that's a good question. Okay, so you were first asking how long these are kind of in time?
Yes, and do you identify when they start and when they end? And if so, how do they distribute across different time domains?
Yeah, that's a good question. So rastermap has no knowledge of the sequences actually. So these sequences are about 10 seconds long, but rastermap, I actually like have run additional analyses on top of this to try to pull out the sequences, because rastermap is basically just looking for clusters that have correlations with potentially positive time lags. And so basically that's all that it's finding here. So yeah, I'm not sure if I'm answering your question well.
So you're agnostic.
Yeah. So the sequences could be potentially more complex. You can see it could be like two sequences happening at the same time. So I've tried sorting, for instance, like the songbird data, and sometimes you'll see those sorts of things happening. But yeah, so it's ultimately how many time lags you use in the data that determines how long in the future it can potentially look for a sequence to happen.
Thank you.
Very quick question from the audience. Alejandro Boria, in your comparison, was UMAP also using a cosine distance function?
That's a good question. I think I did also try that, but I should double check that. With t-SNE, I was using a cosine distance function.
You were, no?
Yeah, so I think I was, but yeah. Great.
Johannes Kappel, very exciting talk, bad jokes appreciated. Can rastermap be integrated into the SUITE2P pipeline?
That's a good question. So right now, SUITE2P has a visualization option, which actually allows you to see rastermap in there. And so you can actually then look at, so it's in the visualize menu in SUITE2P. You can open it up and then you can select neurons in the rastermap and they'll light up in SUITE2P, and you can go in and see where those neurons are in the brain, which might be an additional way for you to explore your data.
Byron Price, when you get 40% explained variance on the mouse neural data, on what timescales are you sampling? I'm assuming it's 2 photon given the number of neurons.
Yeah, that's a good question. So this is sampled at 3 Hertz because we're recording so many neurons. I think this recording is 40,000 neurons. And so the variance explained is at 3 Hertz. So bins of 300 milliseconds. And so we actually have a downsampling step in the neural network from the 50 Hertz data to the 3 Hertz data here.
Another question, how to compute receptive fields of neurons in your final slide? Extract from the natural images or by measuring with sparse noise?
That's a good question. So in this case, it was done with the natural images with just linear regression, ridge regression. So we had enough natural images to do it that way.
Mm-hmm. Another question, how variable are cluster-driven spatial parcellations between subjects, between animals?
Sorry, what was the first?
Variable, how variable?
How variable?
Yeah.
Oh, we haven't really quantified that directly. So yeah, I don't have a good answer for that. We see global and local clusters across neurons, which correspond to retinotopy. But yeah, I don't know at the moment.
It's time, so we can go start with our next session. I'm quite excited about this session because we'll be actually looking at cnidarians, those animals that doesn't really have a central nervous system, this distributed nervous systems. So it's not going to be whole brain imaging, it's going to be whole body imaging, I guess, and whole body nervous system imaging. I'm also particularly excited because we'll have these three speakers, Rafa, Raul, and David, who looked at the ensembles across multiple species. I remember David speaking of flies when I started my PhD, then rodents, and now cnidarians. Same goes with Rafa and David and Raul too. So our first speaker is going to be Rafael Yuste. We'll be hearing about hydra nervous system. Rafa is from Columbia University. Rafa, please, you can go ahead.
Thank you. Let me see, make sure we can see the slides. All right. So why? Let me try to motivate why we're looking at cnidarians. So, and one way to explain this is going to one of the most beautiful experiments that I know of, which was carried out by this trio of people, Francis Crick, Sidney Brenner, and Leslie Barnett, which demonstrated that the genetic code had 3 letters. They broke the code, the genetic code. And genetic code, as you know, is the relation between the structure of DNA, nucleotide structure of DNA, that determines the structure of RNA, and that determines the sequence of amino acids in a protein. So you can think of the genetic code as a series of unidimensional matrices. And if you think of neuroscience, well, you could argue that maybe the critical, the neuroscience is another exercise in cryptography, and that at the end of the day, what we're trying to do is to break the neural code. And the neural code could be the relation between the neural activity matrix, so a matrix also, but in this case, instead of being unidimensional, it has 2 dimensions. So each row would be a neuron, the neural activity of a neuron. It could be 0 or 1 if it's active or not, and each column would be time. And this matrix would capture, as you've seen earlier, in Misha Arendt's talk, the entire activity of the nervous system. And this neural activity matrix determines the muscle activity matrix, in which every neuron here would be a muscle cell or a muscle unit in vertebrates, and every column would be time. And this matrix would determine the behavioral matrix, in which every row could be behavior, one particular behavior, as a function of time again. And a small animal would have a short behavior matrix, and animals like us, where we have compositionality of behavior, could be essentially an endless, an infinite behavioral matrix. So the critical insight that this trio had to break the genetic code was they didn't break it in studying human DNA. No, they went to the simplest animals, simplest that had a genetic code, and those were the phages, bacteriophages. So for the same reason, you could argue that maybe we should try to break the genetic code in the simplest, the neural code in the simplest animals that have a nervous system. And incidentally, the critical insight that this trio got is that the genetic code was built in codons. Sidney Brenner coined the word codon, and the codon was the unit, the functional unit of the code. And the functional unit of this neuronal code could be the ensemble. That's why we're so excited about the ensembles, because the ensembles could be the codons of the nervous system. And we don't know how big they are. But if you look at the evolution of the nervous system, it was actually Sidney who suggested that instead of trying to figure out break the ensemble code in mammals, we should just go to the simplest animals with a nervous system. And these happen to be the cnidarians. So about 750 million years ago, for reasons no one understands, in the tree of life, the metazoans had a branch, and right before that, the first nervous systems occur. The first neurons show up in evolution. And the first branch is called cnidarian. So these are animals that have radial symmetry, like jellyfishes, corals, sea anemone, hydrozoans. And the next branch are the animals like ourselves that we have bilateral symmetry, and that encompasses pretty much all the laboratory animals that you study, from the worms all the way to flies, the mice, or humans. So we followed Sidney's suggestion, and we started to examine the neural activity of a cnidarian, and we chose Hydra vulgaris, which is transparent, is small, about millimeter or so to a centimeter, depending on the species and how much you feed it. Hydra is very interesting. It does not senesce, it doesn't age, and has tremendous regenerative potential. And we use a method that you've heard about, which is called calcium imaging, to measure the activity of all the neurons in Hydra. So these animals have a nervous system which is distributed across the entire body of the animal, from the foot to the head to the tentacles. And this nervous system, depending on the size of the animal, can have from 200 to 2,000 neurons. These neurons belong to 11 distinct neuronal cell types from sRNA sequencing, and they form two sheets of neurons that we call them nerve nets, one in the ectoderm and one in the endoderm. Okay, so with calcium imaging, as you know, we can measure intracellular free calcium in a neuron, and it happens to correlate directly with the pattern of action potentials that the neuron has. So this is work that I did when I was working with Larry Katz, who died and whose memory I honored. And I have to confess to you that we found this by chance. We were looking for a method to record the activity of neural circuits. And we found that if we label neurons with calcium indicators, you could actually indirectly record the spikes of a whole group of neurons. And these Hydra neurons have action potentials, all or none action potentials. And building transgenic animals, we can put in a calcium indicator into practically every neuron in any animal. And with that, we obtain data like this one. This is a movie of the activity of the entire nervous system of one little Hydra as behaving, is contracting and extending. And you can see the individual neurons in white, and you can see when they flash, when they're activated, and those correspond to action potential. The signal to noise is so high in some of these movies that at least for periods of time, we think that we're recording every single action potential from every single neuron. So that matrix of neural activity that I was talking about, we can obtain it at least in certain circumstances for these early metazoans. So what have we found doing this brain activity map, so to speak, of the nervous system of Hydra? And something very interesting happened. So there are neuronal ensembles, just like the one that we were talking about in the mouse and the zebrafish. No? So there's groups of neurons that fire together, and in some cases, like on the left side, they're correlated with behavior. This behavior is called contraction burst. It's a very common behavior in Hydra. So the animals contract, and they do that in a burst. And when that happens, there are groups of neurons, actually an ensemble of neurons that is active at the same time. So they're correlated with contraction. But on the right, you have other ensembles, and this is spontaneous activity. And this was quite a shockaroo. So here's the animal sitting there doing nothing, hanging out. And if you can notice, there are a bunch of neurons that are firing together. No? So this is another ensemble, spontaneous correlated activity between a group of neurons. And we're trying to understand what is this spontaneous activity good for. No? But when you map these ensembles in the nervous system of Hydra, again, we have the luxury that we can map the activity of every neuron, sometimes every spike from every neuron. So we can go to town and find out how many ensembles does this animal have. No? And the answer is about a dozen, about 12 ensembles, more or less. No? And the one thing that's very interesting is that these ensembles are non-overlapping. So what that means is that even though they're intermixed in the body of the animal, like getting these two nerve net into sheets of neurons, each neuron belongs to either one or another ensemble. So if you actually, in this correlation matrix, there's essentially no overlap between ensembles. They're completely independent from each other, at least in terms of their neuronal activity. In some of them have to do with contractions, with behavior, other behaviors like nodding, tilting of the head, longitudinal contraction, radial contractions. But again, some of them are spontaneous, other ensembles looking for function. And maybe these little first metazoans with nervous system are thinking. I don't know what they're they're thinking about, but maybe that the spontaneous activity could be some sort of integrator of information. So when we were building these transgenic animals to measure the activity of every neuron in Hydra, by chance, we got the transgene into the muscle cells, and in fact, into all the muscle cells of the animal. Hydra has two sheets of muscles, one in the ectoderm and one in the endoderm. And before we knew it, we're taking movies like this, where now we're looking at the activity of every muscle cell in the animal, not the neurons, the muscle cells. And you can see how exquisite it is that when the animal is trying to move its tentacle, you can actually see the activity of individual muscle cells. And of course, when it contracts, you can see a massive activity of the muscles, both in the ectoderm and in the endoderm. Let me show you another movie. In this case, this is a Hydra that's budding. Hydra have both sexual and asexual reproduction. So an example of asexual reproduction are these 4 buds that are growing out of the mother. And these 2 buds here are actually older, and they are behaving independently of the mother. They have this muscle activity that's synchronous, that's individual. It's independent from the mother, but the little buds that are the ones that are just been emerging here are behaving as is part of the mother. So this is fascinating because you can literally watch the emergence of an organism in terms of the nervous system and in terms of the muscle activity. So what have we learned with this muscle activity? We've obtained this muscle activity matrix that I was telling you about. So what we've learned is that there are different patterns. This would be the equivalent to the eigenvalues that we just heard about of the matrix, different patterns of muscle activity, spatial-temporal patterns. So these are the main ones. No? And some of them correlate with one behavior, some of them correlate with more than one behavior. Okay. And here we're plotting on the top the activity of the ectoderm, and at the bottom the activity of the endoderm. We did this with 2-color calcium imaging, sticking a RCAM in one in the endoderm and the GCAM on the ectoderm. And on the right, you see the chemographs, which are essentially the spatial-temporal representations of the activity of this movie that you see, whose frames you see on the left side, just to give you a feeling for the richness of spatial temporal patterns of muscle activity. Here you, for example, these little lines here correspond to contractions. And you can see this contractual burst that I was telling you about here. And one of the discoveries, I mean, this is fascinating to study an animal like Hydra, where essentially nothing is known. So we encounter discoveries left and right, and that reminds us what's so exciting about science and biology, to go in and find new things. No? And one of the things we found was very surprising is that the same muscle cell can be active in different behaviors, okay? And sometimes with different time courses, and this is something we were not expecting. So this could explain why an animal that is so simple in its structure could do a lot of different things, because it shows multifunctionality in terms of its muscle cell. So the muscle cells can, they're like jack of all trades. They can be used, the same muscle cell active in a contraction pulse when only the tentacle is moving, when the animal is nodding, when the mouth is opening, and sometimes it does it, like it shows here, with faster kinetics and sometimes with very slow kinetics. So this is something that you'll hear later is part of the model that my colleague Adrienne Fairhold and her lab have done on these animals. So let's talk a little bit about the behavior. So I told you how we can obtain the neural activity matrix and the muscle activity matrix. And how about the behavioral matrix? Well, you can put these animals under the camera and use machine learning or deep neural networks to essentially classify all the behavior that they do for hours. And these are some of the simplest behaviors: contraction, elongation, the tentacle swaying. But there's some fascinating behaviors, like here on the left. This is called somersaulting. Because these animals have only one foot, if they want to move from the bottom to the top here, if they live in freshwater ponds and rivers, if they release the foot, they get pushed around by the current. So what they do is they somersault by attaching the head to the substrate, and then let go of the foot. They flip the foot over and now let go of the head, and they continue, and then they attach the head farther up, and they continue to do that. They somersault, and you have to realize these animals have a few hundred neurons, and they can somersault without the centralized nervous system. They don't even have a ganglia. No? Another form of locomotion is inchworming. So it's a little bit like a somersaulting where you don't flip over. You essentially stretch yourself, you attach the head, you let go of the foot, you contract the body, and then you essentially do that. And then there's also some feeding behavior, which is also quite sophisticated. Again, think that these animals have a few hundred neurons, and look what they can do. No? So you put some shrimp on the bath, and they extend their tentacles, and using cnidosis, which are characteristical of cnidarians, they're supersonic harpoons that inject toxins. That's why the jellyfish sting. All cnidarians sting. And they catch these shrimp, they paralyze them, and they swallow them. No? And they do that with an incredible efficiency. You cannot help look at this movie and believe these guys are intelligent. There's look at this little shrimp, the last one, boom, it just caught it. No? So this is an example of the types of behaviors that they do. And with this similar method to the one you've heard before, TSNE, both with supervised and unsupervised learning, we've classified all the behaviors of the animals, really behavior animals, in the lab, and study how this behavior changes when we modify environmental conditions. And to our surprise, behavior is extremely robust. No? So this relates also to the first talk of the meeting on control theory. You know? So we think that this nervous system of Hydra somehow has figured out a way to do distributed control, because the animal more or less does the same thing, whether you put it in the dark, in the light, whether you feed the animal a lot or not, whether it's small or large. There's some differences, minor differences, also if you compare different species. No? So it seems as if these animals have been programmed by nature with some sort of control algorithm that maintains the behavioral repertoire robust, at least for these basal behaviors of Hydra. So, so I'm showing you how now it's experimentally tractable to take an animal, an cnidarian, the ones with the simplest nervous system, and to try to break the neural code by measuring the neuronal activity matrix, the muscle activity matrix, and the behavioral metrics. And you can imagine doing some dimensional reduction, like just heard about, finding the latent variables of these metrics and linking them in a series of equations in a computational model. And this is what my collaborator, Adrian Fairholt, is trying to do. And the first step is a work by her postdoc, Hengyi Wang, is to build the biomechanical model of the contraction burst, this behavior that I showed you earlier. So they build a model in which they have a nerve net with endodermal and endodermal components. And these neurons are connected to the muscle cells, which we call them epithelial, because the muscle cells are also the epithelium. These animals are in the beginning of times, the muscle was the skin. It's the same. The skin muscle was the same thing. And putting in equations that govern neural activity, that govern calcium dynamics in these muscle cells, and coupling those equations to a set of equations that govern the biomechanics of this hydrostatic skeleton. Hengyi and Adrian have been able to reproduce in silico this contraction burst. And this is an example of a in silicon Hydra that is moving around and contracting and swinging, as you've seen with the real Hydra. And here on the top, you see sort of the activity of in this case is the muscle network, but you can imagine plotting the neuronal activity as well. And this model actually reproduces very closely. The model is in blue. The real behavior is in red. You can see how this is a good fit to the model. It doesn't mean that this is how the animal works, but at least it provides evidence that you could use a computational approach to try to break this neural code. This is just an example of the kinds of things that you could do with Hydra. And of course, the goal is to go to town and explain how all the 12 ensembles work and what do they do. And just to finish, one of the advantages of Hydra is that still gives me shivers when I say that. You can take the animal apart into individual cells, dissociated cells, and then it puts itself back together over a period of a few days. So here you have it on the left. We've taken the animal apart. This is one of the animals that had GCAM in all the neurons, and you can literally watch how a nervous system gets put together, how the whole animal is put together in front of the microscope. And eventually you end up with a completely normal hydra. And we found that the neurons initially, when the animal gets reaggregated from this dissociated soup of neurons, the neurons fire individually, and eventually they assemble into modules. And these modules are the proto-ensembles that later became distributed across the whole body. So this would be a wonderful preparation to study how ensembles develop, how they put together, actually how an animal can self-assemble. So with this, I will finish. Sidney Brenner is the person that recommended us to work on Hydra. He was my mentor in Cambridge, and the work in my lab has been done by a very talented group of students and postdocs that I'm listing here. We've been funded by federal funding agencies of the US, and this work has also stimulated a series of collaborators across the country and the world that are now working on Hydra. So this is becoming a little, this is becoming a thing, no? And this is some of the people around the country and in Paris as well that are working on Hydra. And I just wanted to finish with a little plug for Oliver Hover's lab. He's also interested in working on Hydra. He's a colleague at Columbia, and we're jointly recruiting a postdoc. If you're interested, please write to me or to Oliver. Thank you.
Thank you, Rafa, for this wonderful talk. For those who has questions, please just use the Q&A button for asking your questions. In the meantime, I see Rosa raised hand. Rosa, please go ahead.
Thank you, Rafa, for a great talk. I was obviously fascinated by the last part of the reaggregation, or whatever you call it, of the Hydra, yeah. and also about these contraction bursts you described at the beginning, and which to me are reminiscent of the muscle twitches that are observed in many developing species. So my question is, do you see, when do contraction bursts emerge during the reaggregation of the Hydra?
So they emerge pretty early, about 48 hours. You can see these reaggregates that have neuronal patterns that are just like the contraction burst. And then you can start seeing contractions of the reaggregates. And within a day or two, you can see them going through like the burst of contraction. And this is right at the time in which they essentially decide on an axis of the body, a body axis, and that's when they grow the tentacles and they form a proper animal. But it's fascinating that you mentioned, I'd forgotten that the twitches happen also with in mammals early on. But let me tell you that we don't understand why the animal has this burst. I mean, we understand that the animal contracts. Every animal wants to contract once in a while, but why do it so quickly and so frequently in this burst? Mm-hmm. So we're suspicious that maybe the reason has to do with water excretion. So cnidarians are so simple that they don't even have organs. They have tissues, but no organs. So they don't have kidneys or nephrons. So, and this Hydra lived in fresh water, which means that they have a huge osmolarity problem, because if you put an animal in fresh water, including ourselves, at some point our cells are going to burst from osmotic shock. No? Mm-hmm. And one way to solve this problem in Hydra is to squeeze all the water out, just like you wring a towel. And we think that this contraction burst is like the animal getting rid of all the water. It's just a hypothesis. Mm-hmm. Yeah.
Thank you. There's a question several questions from the audience. Anita Devineni asked a question that I was also thinking, so I'll go ahead and shoot that one. What defines the 11 neural cell types that you mentioned? Gene expression, activity patterns, and then when Hydra reassemble after dissociation, does this include differentiating into these different cell types, or there's some other principle exists?
great question. So the number of 11 cell types that's come out of a transcriptomic study that my colleague Selena Giuliano, who's at the University of California in Davis, has done. So Selena and her team have actually done transcriptomic with not just every neuron, with every cell in the animal, and using t-SNE precisely, classifying the neurons into these 11 clusters, and they correspond to groups of neurons that are part of the ectodermal or endodermal network, and in some cases, these clusters correspond to the ensembles that we're talking about. So there's at least one of the clusters that is exactly one of our ensembles. So when you dissociate the animal into individual cells and the animal starts to put itself together, that happens without having to differentiate new cells. So this is not regeneration in the sense of making new neurons or new cells. This is using the same cells that you just put together, the ones that are still alive. Then they find themselves and they go back to what they were, no? And so, I mean, there's a this is a very special type. I was a very powerful type of self-assembly, and we really don't understand it. So it's a fascinating phenomenon. It's been described by Gierer in Tübingen already almost 40 years ago, and it's just as far as I know, the biology in general hasn't really tackled this issue, and this is a challenge for us to understand how you can get away and do that. No? Yeah.
I will combine a few different questions into one question. That is, when you look at these ensembles of cells, do you find like hub cells or cells that are completely not belong to an ensemble? What kind of structures you see within the ensembles of Hydra?
Yeah. So we don't know yet if there are critical cells that could be hub cells, trigger cells, or pattern completion cells. We don't even know if these neurons are excitatory and inhibitory. I mean, we do know that they have action potentials, that's for sure. No? But we know very little about these ensembles. There's occasional cells also in our movies that do not seem to correlate with an ensemble, but the large majority of the cells are part of an ensemble.
I see Misha has a question. Misha, do you want to turn on your microphone and ask?
Yeah, related to the earlier question, so they don't become different cell types when they reassemble, but if cell types can take part in different functional ensembles, is there any kind of functional reconfiguration after reassembly?
Yep.
Do you know that? Yeah?
Unknown. One thing that happens in Hydra is that these animals are in constant regenerative mode. So they have a lot of stem cells that are continuously generating not just neurons, all the cells in the body. And that, in fact, they live in a state of eternal youth. They're supposed to be immortal. I mean, you cannot prove that someone is immortal, of course, but the same animals have been studied, maintained alive by my colleague Daniel Martinez at Pomona College. And over a period of 12 years, he saw no sign of senescence or age-related phenomenon. And for an animal that small to last 12 years, if you just scale it up, it will be equivalent for a human to last 10,000 years without any signs of senescence. So it is likely that this continuous regeneration allows them to essentially live forever. I mean, you can always kill them, but in principle, they're good to go. No? And within this continuous regeneration, we presume that all these cell types are getting replenished. They're going to be made continuously. And in fact, in the t-SNE plots of the transcriptomic, you can see the sort of trajectories of cells that correspond not just to isolated islands, but to more like processes of differentiation.
So there's actually many questions, but again, I need to select some. I see Emiliano Jimenez Marquez ask, perhaps I should first ask, do they learn or do they exhibit any sorts of plasticity? Yeah. And if so, if you were to crunch a Hydra and reassemble, can they still keep some memories of their experience?
This is a fantastic question. We're studying learning in Hydra, and we've found, actually a postdoc in the lab with Taro Yamamoto has found that they can habituate there's a feeding response. I didn't go into details, but you can trigger the feeding response of Hydra, the one in which they go with the tentacles and paralyze the prey and swallow it. If you put an amino acid on the bath with glutathione, somehow amino acids means food and means shrimp, and that triggers the whole behavior. It's like a fixed action pattern. Again, they're doing it with like 300 neurons, so it's unbelievable. If you keep putting the same amino acid over and over again, eventually the Hydra learns that this is no shrimp and they stop responding. So this is a form of habituation that we're trying to study. But the second part of the question is something that is also fascinating, is learning cell autonomous. You can dissociate the animal, put it back together. Does the animal still remember that it's habituated? This is a great experiment. Yeah, we haven't done it, but it should be done, yeah.
Thank you, Rafa. You have so many questions in the chat, so you'll have to go and answer them one by one. But right. I would suggest, for the time's sake, we should move on. Thank you again for this fantastic talk.
You're welcome. Thank you.
So our next speaker is going to be David Anderson from Caltech. And again, just like all the other speakers, I remember when I first started my PhD in 2003, David was giving talks on flies, then amazing talks in rodents, and now we're hearing David also giving amazing talks with jellyfishes. So today we're going to hear about that work. David, please go ahead.
Thank you. It's a pleasure to be here at this exciting meeting, especially to be able to talk in a session on cnidarians. Let me make sure that everyone can see the screen. Can you all see this? It's in presentation mode. Is that okay?
Yes, we can see.
For the last 20 years or so, my laboratory has been working on understanding neural circuits that control components of internal brain states that are expressed behaviorally by emotional behaviors. We're not concerned with the issue of feelings. That's something that can only be studied in humans because it's subjective, but we are particularly interested in the question of the evolution of these internal state control mechanisms. How and when did they develop on the planet? We all have our own intuitions about what animals have and don't have emotions. We are happy to attribute them to animals that we are familiar with, but that anthropomorphism breaks down when we look at animals that are much less similar to ourselves in their morphology and their behavior. And there it becomes a sort of subjective question that depends on one's underlying assumptions and preconceptions. Now, one way of dealing with this was Darwin's approach, which was to simply assume that all animals had emotions. And he famously wrote that even insects express anger, terror, jealousy, and love by their stridulation. But that's just a belief. It's not knowledge, and it's anthropomorphism, and one can be fooled. And so it's possible that animals that may look like they are showing emotional behaviors may simply be very fancy, highly tuned robots with no types of internal states, the kind of thing that you would build if you were building a fancy Land Rover from Mars. So as I said, one can be fooled into thinking that behaviors that may superficially look like they are emotional are in fact simply robotic reflexes that are very sophisticated. And this was brought home, this concept was brought home strongly to me when I read an influential book by the cyberneticist Valentino Breitenberg. And Breitenberg described how it was possible to wire up a series of robots that could, by simply having sensors for particular signals wired to motors and wheels so that they could drive or inhibit them, that according to the sign of the effect on the wheel motion and whether the connections were ipsilateral or contralateral, you could make these animals head towards an object, away from an object, etc. And in a not so subtle trolling of psychologists, he labeled these robots as exhibiting fear, hate, love, curiosity, etc. And so that really, really brings home the question of the need to examine animals to see whether they are built this way or whether there are internal states that drive their behavior, and if so, how these states are controlled. And over the years, there have been many studies in many laboratories, in many organisms, including Misha Ahrens in zebrafish and my lab and others in flies and mice, showing that there are internal states with properties like persistence and scalability and valence and generalizability that control behaviors. But we were interested in the question of whether there are any animals on the planet that actually do behave like Breitenberg vehicles. And if you were going to look for an animal that is a prototypic biological Breitenberg vehicle, you would look for the most primitive animal that has a nervous system, and that would be a jellyfish. Now, one could also do this in Hydra, of course, as Rafa described, but there are a couple of features of the jellyfish that made them particularly attractive to us. And for this species, we chose a cnidarian zebrafish called Clitia hemispherica, which had been introduced to biology by Evelyn Huliston and used to study early steps in oogenesis and development. And Evelyn and her group works in the marine lab at Villefranche-sur-Mer, and she was very kind in 2018 to invite a postdoc from my lab, Brady Weisberg, to come and spend 3 months there learning how to work with the jellyfish and culture them, and to establish a very important collaboration with Suyo Momoza, a molecular biologist who works at the laboratory. So Clitia are hydrozoans, which have a medusa stage, a jellyfish stage. They have a sexual reproduction cycle where eggs and sperm fertilize and make a free-swimming larvae called a planula that sits down and metamorphoses into a colony of polyps of two types, one that eats, which is sort of like Hydra, and another one that produces polyps, which get spit out into the water to complete the life cycle. And interestingly, Clitia is a member of Hydrozoa, the same group that contains Hydra, and is one of several different clades of jellyfish species, including Cubozoa and Scyphozoa. And the ancestral form of Hydrozoa is believed to have had a medusa stage. And Hydra is believed to have lost the medusa and planula stage. And in fact, there are a number of examples in phylogeny where the medusa stage has been gained and lost repeatedly by hydrozoans over evolutionary time. So Clitia are small. A large adult is upwards of half a centimeter at most. And the baby ones are considerably smaller. It has a characteristic jellyfish structure with nerve rings surrounding an umbrella, striated muscle at the rim, smooth muscle, and then a subumbrella nerve net, as well as nerve nets in the ring and in the tentacle bulbs and in the nematocysts. These jellyfish show a variety of interesting behaviors. They show swimming in two different modes, one in which their tentacles are extended, which we call the hunting mode, one in which the tentacles are contracted and the swimming rhythm is very different and more regular, which we call an escape swimming mode. They show various types of defensive behavior against mechanical, noxious mechanical or chemical stimuli, including crumpling and freezing. They show complex feeding behavior, and they also lay eggs. And they seem to do all of this through a series of autonomous modules, raising this general question that Rafa referred to as how such autonomous modules can be brought together to exhibit coordinated organismal behaviors in the absence of any central ganglia or central brain. Now, Clitia has already had a number of advantages to it as a model system for genetics in particular. And our objective was to find a system that was good not only for imaging of neural ensembles in adults, but where we could flexibly and readily perform genetic manipulation. So the entire life cycle of Chlamydia is established in the laboratory from sperm and egg all the way through to sperm and egg again. It takes about 6 to 8 weeks. They have sexual reproduction, allowing you to do genetic crosses. And since I'm a geneticist and a molecular biologist by training, the ability to do such genetic crosses is for me a sine qua non of a truly genetic organism. Its genome has been sequenced. We've recently generated the cell type atlas by single cell RNA-seq, and CRISPR-Cas9 knockouts have been established by Tsuyo Momoza. But at the time we entered the field, there was no system for transgenesis. And that was the major thing that Brady and Tsuyo set out to do. And it was not obvious that this would work. There were many, many failures, and it took close to 3 years to get transgenesis to work. The way that it works is that you get the animals to lay eggs, females or males, to produce sperm in the lab. An advantage of this system is that a female, when it's triggered to spawn, can lay thousands of eggs that are available in a day. And so you can literally inject hundreds and hundreds of eggs per hour. And if you need to generate 1,000 injected cells in a day, you and a friend can do it. The eggs are directly microinjected with DNA constructs. Tsuyo developed the TOL2 transposase as a protein to facilitate integration into the genome. There's fertilization. We then do in vitro fertilization. So these are test tube jellyfish babies. We wait for the larva to form. We check for expression of an M-Cherry reporter in the larva. We allow them to sit down and to form polyps and to screen at the polyps for transgenic animals. This is in the F0 generation, and you see that there is mosaicism, and eventually about 30% of the polyps that survive and grow and form polyp colonies produce jellyfish, F0 jellyfish, which are transgenic. And fortunately, essentially 100% of those adult transgenic F0 jellyfish, when crossed to the next generation, are able to produce F1 progeny that are non-mosaic. And what that means is that you can perform those crosses in other genetic backgrounds, for example, or bring other transgenes into the system very easily. So if you wanted, for example, to have a muscle promoter driving one reporter and a neural promoter driving another reporter, you could easily do that. And these lines can now be maintained indefinitely as propagating polyp colonies in the same way that Hydra can. So they are effectively immortal, but it also generates clonal experimental non-mosaic F1 animals, and then these can be further bred into the F2. So this system, once we got it to work, has a lot of advantages as a transgenic system. So we decided to focus our initial applications of this technology to a very basic behavior in Chlamydia. In this case, it's feeding behavior. And feeding behavior in Chlamydia is quite interesting. As you can see here from these paired video frames and drawings, a shrimp is trapped by a tentacle and retracted to the rim of the umbrella. And that part of the umbrella is then asymmetrically and vectorially folded inwards to transfer the trapped shrimp to the mouth or manubrium, which simultaneously points in the direction of the infolding umbrella. And after that occurs, the food is transferred to the manubrium where it's ingested and digested, and unfolding occurs. And I can show you what a movie of this looks like here. We've injected a hapless Artemia into the dish. It's entrapped by a tentacle, and the tentacle retracts, it brings it to the edge, and then you see this transfer and infolding of the mouth, the transfer of the fish into the mouth from the infolded umbrella, and swimming continues. So we did a number of experiments. Brady did a number of experiments and found that this behavior can be performed, first of all, in response to shrimp extract, as well as intact shrimp. It can be performed in the absence of a mouth. So you don't need the mouth for the folding to occur. It can be performed in the absence of tentacles as well. And these and other experiments have shown that the whole feeding process is comprised of a series of actions that are carried out by these independent modules, each with their own neural networks: tentacles, ring, and subumbrella, and the mouth of the manubrium.
There was a question about, did you observe any differences in evoked versus spontaneous activities in an ensemble?
So when we do the compare the correlation plots and we do the generalized linear models, we don't get a perfect prediction. We get about 0.85. So there clearly are some differences between evoked and spontaneous activity. However, importantly, the performance of that GLM is no worse in predicting evoked when trained on spontaneous than it is in predicting a held out data set of spontaneous activity when trained on spontaneous activity. So for that reason, we think that any differences that we see reflect the underlying apparently stochastic variation in the ensemble membership that occurs even in spontaneous conditions.
Mm-hmm. Actually, I see Raul is back, but I would like to read this question about fed versus starved animals. Do you see differences in explorations in these, like the way these animals move, they sort of cover ranges?
Yeah, that's a great question. We've identified two different modes of swimming. In one mode, the tentacles are extended and the jellyfish sort of erratically and kind of lazily contract their bell. And in the other mode, the tentacles are retracted and there's a very regular pulsatile pattern. And so there are clearly two swimming states. We haven't tested whether they're affected by starvation, but obviously we're very interested to gain control over those states so that we can study how they're encoded.
Thank you, David. I see that Raul is back. So Raul, go ahead and share the screen and let's give it a go again.
Its first part is about jellyfish behavior. And the second one is about drifting assemblies in bilaterians. So let's get right into part one. So here we were developing a model which bridges the scales from single neurons to the behavior of jellyfish. So the goal was to develop a comprehensive model bridging single neurons, the nervous system and the muscle system to the swimming behavior of scyphozone jellyfish. So the motivation is that jellyfish are comparably simple and actually allow to directly relate neuronal dynamics and behavior, as we have already heard in the previous talks. They are evolutionary old, so they tell about the origins of nervous systems. As we have already also heard, they are our most distant relatives with homologous neurons, but they already have spikes and they communicate with chemical synapses. So it's interesting to study them. And the swimming of jellyfish, which has not been addressed in the previous talks, is interesting also for engineering, because jellyfish are highly efficient swimmers. So they are studied a lot in the field of engineering, and they are becoming a dominant taxon in the oceans. So it's also worth to study them. Yeah, what we were focusing on was Aurelia aurita, so a scyphozone jellyfish. Before the two previous talks, they focused on hydrozones. And they have completely or largely diffuse nervous system. So they don't have the ring which we heard about in the previous talk. The only places where there is some apparent concentration of neurons are the so-called tropelia, which act both as pacemakers and which process sensory information like light and gravity. The Rhopalia, they stimulate the motor nerve net, and this then, which is a nerve net distributed over the subumbrella, so the lower part of the jellyfish, and this then generates these nice swimming pulses. The diffuse nerve net, which is an RF-amide, which consists of RF-amide neurons, which we heard about in the previous talk. Actually, this is considered to at least in parts steer the turning via radial muscles. So the schematic setup of the subumbrella is now as follows. We have the motor nerve net distributed here over the subumbrella, except for a gap where the mouth of the jellyfish is. Then we have the radial muscles here, which steers this constriction, which then in turn leads to the swimming movement. And we have the DNN, which extends over the entire subumbrella and also over the rest of the bell, with the exception of the mouth. And this excites so-called radial muscles here in the rim of the jellyfish. Our approach of modeling this nervous system is as follows. So we just take the Rhopalia for granted, and we assume that they excite the motor nerve net and the diffuse nerve net, which then excite their respective muscle systems, which then changes the bell shape. The change of the bell shape acts back on the muscles because the muscles' strength changes depending on the elongation of the muscles. And of course, the bell shape then, interacts with the fluid. So we had to start by modeling single neurons. Fortunately for the Scyphozoan jellyfish, there is some knowledge, some electrophysiological knowledge on the single neurons. They are spikes are generated by sodium or and likely also potassium channels, so like in bilaterians. The neurons are coupled via chemical synapses, which are actually, interesting difference, bidirectional. So there are vesicles and receptors on each side. So there is no real presynaptic and postsynaptic side. This is how a spike looks like in a Scyphozoan jellyfish. And this action potential here, spike, generates then depolarization in the postsynaptic cell, which is strong enough to generate also a spike in the postsynaptic cell. So this leads to through conduction in the nerve nets. Whenever you stimulate a bit, then the activity goes through, and in contrast to hydrozones, which we heard about. So yeah, I was just saying that here the global activity was not directly observed yet, in contrast to hydrozones, as we have seen in the last talks. And so we now, we started with modeling single neurons. And we used a biophysical Hodgkin-Huxley type neuron with 5 transmembrane currents. It was known that these 5 transmembrane currents are relevant, and we could fit we could determine the channel kinetics by fits to already existing voltage clamp experiments, and furthermore we could determine the membrane capacitance and the synapse model by the EPSP shape, the action potential inflection point, and the time to the action potential peak. So this is how an action potential spike looks like in our model here. And what catches the eye is this interesting bump here. And this is a consequence of the synaptic reflux. So as I said, the action potential generates a neurotransmitter influx into the synaptic cleft, but there are receptors on both sides. And actually this here is the EPS, the result of the EPSC on the presynaptic side, which generates this little bump. And if one looks at this at the experiments carefully, then one notices, oh, there is also this bump, for which we now have an explanation. There are also several experimental predictions coming from the single neuron measurements. For example, the time course of the currents during action potential, the importance of currents for the refractoriness of neurons, and also that we expect a long recovery from inactivation to explain the very long refractoriness observed in the experiments. So now let's continue with the network, the NerveNet. So, as I said, we focus on the sub-umbrella, and there in particular on the motor nerve net, which is here at the inner part of the sub-umbrella, except for the mouth opening. And we model individual neurons just by lines, because they look really like this under the microscope. They have the soma, and then there are two protrusions. There's no distinction between axon and dendrites because the synapses are just symmetric, and these protrusions are 5 millimeter long, so really long. And this we incorporate in our model, and the geometry is covered by incorporating the signal conductance time. So we assume that the time for the transmission of an action potential from one neuron to the other depends on the distance of the soma to the synapse, and then of the synapse to the other soma, as you would expect when the action potential travels here along. Interestingly, there are more quantitative facts known about scyphosome nerve nets. For example, the preferred orientation of the neurites. Namely, the neurites are preferentially radial near the rhopalia. They are preferentially tangential between the rhopalia, and they do not have a preferred orientation near the mouth. We model this using a von Mises orientation preference distribution, and you can compare here the von Mises distributed network with a random, completely random network. For the DNN, there is less quantitative data, so we just use random orientation preference. What is known is that the neurites are much shorter, which you can see here. And of course, that the DNN extends into the rim. So this would be the DNN and the MNN on top of each other. Already the geometric setup of the nerve net yields an interesting experimental prediction, namely on the number of motor nerve net neurons in a scyphosome jellyfish. This number is not known, but what is known, what has been measured, is the average intersynaptic distance. So the distance between crossings of neurites, and basically at each of such crossing, there is also a synapse. This has been observed. If we now measure the same quantity in our model networks, in our von Mises model networks, which are the biologically most realistic, then we see, yeah, the average intersynaptic distance decreases with neuron number, as you would expect. And it reaches the 70 microns measured at a number of about 8,000 neurons. So these simulations are for jellyfish of size 4 to 5 centimeters, which is our prediction then for the number of neurons in them. We can also, of course, that's what we did it for, combine the single neuron model with a network model and basically connect the single neurons to a network with the already mentioned topology. And now, if we assume that this rhopalium here on the right-hand side stimulates the motor nerve net, then we see that there is a pulse of activity spreading along around the network here. It spreads and arrives at the other side. At some time point, the entire process is below 100 milliseconds. And this yields a different prediction for the number of motor nerve net neurons, because the delay between opposite sides has also been measured to be on average 30 milliseconds. And if we compare this with the delay in our networks, here we need to simulate jellyfish which are 3 or 4 centimeters large, because the experiments were done with jellyfish of 3 to 4 centimeters size. Then we see that, well, again, we reach the experimentally measured delay at around 8,000 neurons. The gray labeled here is the standard deviation of the experiment. So we also see that actually the von Mises and the uniformly random distributed networks do not have much differences in the delay time. But if we look for the same number of neurons, like 8,000 here, which gives the same delay time in both networks, there are more neurons because the average intersynaptic distance is smaller, so there are more neurons in the uniform network. So this von Mises distribution may be an evolutionary adaptation to save synapses. It may also just be a consequence of the development of the Aurelia jellyfish from an Ephira. Now we continue to modeling the bell, the muscles, and the behavior. So we model the bell as an elastic object with mass points connected by dampened springs. We put the entire thing into a simulated hydrodynamic environment using the so-called immersed boundary simulation methods. We model the effect of radial muscles as a force towards the center line, and the muscle contractions are due to the neuron spikes, due to the spikes of the motor nerve net. Each spike generates a little muscle twitch, and these then sum up to nice contractions. These contractions look as follows in the simulated hydrodynamic environment. So here the rhopalium is initiated on the left-hand side, and we see that the jellyfish contracts, as we also know it from real jellyfish. The coloring here is the vorticity of the fluid, so how whirly it is and in which direction the whirls go. And we see this contraction and the forward propulsion of the jellyfish. If both DNN and MNN are activated, then the activation of the radial muscles at the margin of the jellyfish lead to a turn here in this simulation, which is also in accordance with the assumption what the function of the DNN is in locomotion. So to conclude part one, I've shown you a model from single neurons, nerve nets, muscle, bell, hydrodynamic environment of jellyfish behavior. If you want to know more and read about some details, this has been recently published also. So the second part, as I already said, this will bridge towards ensembles of neurons in bilaterians. We asked, how can assemblies of neurons, neuron ensembles, which are strongly connected, drift, and how can behavior nevertheless stay invariant? So this is motivated by basically 4 observations. So the first observation is that representations change due to adaptive learning, but representations also change spontaneously without affecting behavior. So behavior stays constant. And this has been observed for long-term memories, for odor representations at Columbia prominently, for place cells, location, and goal-selective cells. Furthermore, weights and connectivity change due to neuronal activity, but weights and connectivity also change spontaneously. And where it has been compared, it has been observed that the strength is actually similar, and it affects the excitatory weights, the inhibitory weights, and there is also synaptic turnover, spontaneous synaptic turnover. So the general idea of our model is as follows. So there is an assembly layer here, and the assembly layer processes the input, which is here the image of a nice apple and generates or initiates a reaction, grasping the apple because apples are tasty. Now we ask, how can the assembly in this layer here change? So how can neurons join the assembly like this neuron here? And how can neurons leave from the assembly? And how can this changes be compensated by the input and output neurons? So the input and output neurons should still make strong connections only to the assembly neurons. So they should lose here these neurons which are not part of the assembly anymore. And they should gain connections to the appropriate connections to the new neurons. And this should go on even if, or ideally, we think, even if the assembly has completely remodeled. So how can this happen? This was our question, and we addressed this question with spiking network models of several types, leak integrated fire, Poisson, and binary neurons. We choose just a simple symmetric STDP rule between the excitatory neurons, motivated, for example, by the hippocampal region CA3, where STDP is observed to be symmetric. And there is additional homeostatic plasticity, which implies that the total synaptic input and output strength are fixed. The network is set up as follows. As I said, we consider assemblies, so ensembles of neurons which have strong interconnections. So this is assembly 1 here, colored in blue, and consider neuron 1 of this assembly, and neuron 1 has a strong connection to neuron 2 in the assembly, say to this neuron, that would be this deep blue color, to neuron 3, and so on. So this is, so the deep blue color signifies the strong weights. And also there are input neurons here in green and output neurons here in red, which are in our model, in our simple model, specific to this assembly. So this input neuron here, the green one, this generates a strong input to neuron 1 because it's an assembly 1 input neuron. So that would be here, but it doesn't generate any strong synapse to neuron 31 because neuron 31 is part of assembly 2, and these neurons are connected to other neurons. And the same for the output neurons. So there is a strong input, a feedback, strong feedback input from output neuron 1 to neuron 1, and also neuron 1 projects strongly to neuron 1 here, and there is also a strong feedback output to this green neuron. If we simulate these types of networks, we observe reactivation of assemblies in irregular random manner. So here, these are the reactivations, the stripes here. Apart from this, there is asynchronous irregular dynamics. So the voltage just fluctuates randomly or noisily in example neurons. And now, after 27 minutes, we observe these stripes here in the weight matrix. And let's have a look at neuron 64, which has generated such a stripe. And we see that neuron 64 now receives strong input from assembly 1 and also from the input and output neurons from assembly 1. And it also generates strong output to the assembly 1 neurons and also to the input and output neurons of assembly 1. So this means that neuron 64 has switched from assembly 3 to assembly 1. So perhaps it is this neuron here, which is new to assembly 1 and which was before to assembly 3. And if you look carefully at the spike trains, you will also see, neuron 64 doesn't spike anymore together with reactivations of assembly 3, but now spikes together with assembly 1. So this neuron has completely switched assembly. And if we continue this simulation, then the weight matrix looks completely scrambled and there's also not much structure anymore in the weights. However, we can now re-index the neurons. We can now say, okay, neuron 64 now belongs to assembly 1, so let's call it neuron 31. So let's just re-index it and put it here. So then this cluster looks nice again, and these stripes are all concatenated to this square here. And we can, and if we do this, and if we also do this with this apparently completely scrambled matrix, we see, aha, the assemblies are actually conserved, only they consist of different neuron ensembles. If we put the sorted, resorted, re-indexed weight matrices here on the bottom, we see the assemblies are always there, and it is always the input and output neurons of assembly 1 which attach to assembly 1. So the input and output neurons of assembly 1 indeed follow assembly 1 faithfully, which was our condition for preservation of memory despite the drift of the memory representation, despite the drift of the assembly representing the memory. So analysis and a careful study showed the following interpretation of these dynamics. We have seen that the assembly membership means for a neuron actually that it is in a potential and noise well. So it experiences very little noise if it's in an assembly, plus if there is some weight change, then the weight change basically usually reinforces its attachment to the assembly, which is the potential, well. And only weight fluctuations due to noisy spiking or due to spontaneous remodeling, as observed in experiments, can occasionally lead to the transition of a neuron to another potential and noise well, to another assembly. And the input and output neurons, they do not do these transitions because they experience less noise in our models because they have just a little bit weaker STDP. Of course, they need STDP to be able to follow the assembly. We can also compare our findings with experiments. And there's especially one experiment which fits really nicely. It is a fear conditioning experiment where at day 28, the fear memory representation was labeled, and also at a previous time. So for example, at day 14, at day 7, at day 1, and directly after fear conditioning, the fear memory representations was labeled in a different manner. And then the colleagues looked at a different, at the double staining, and they saw that the fraction of double stained neurons decreases the longer the temporal distance between the second staining at day 28 and the first staining was. If the staining was at day 28, the second staining, and the first staining was at day 14, then there was quite some overlap, but there was a smaller overlap if there were 3 weeks between the stainings, and even more here if there was 4 weeks difference basically. And in our model, this translates to a loss of overlap between the fear memory representations. So the overlap, so the neurons which are part of the fear memory representation, or in this more detailed model, at day 28 and of the fear memory representation at day 14 is still quite large, but the overlap between the memory representation at day 28 and at day 7 is smaller, even smaller between the two neural ensembles forming the fear memory representation at day 1 and day 28 and so on, due to the drift of the fear memory representation. So to conclude, I have shown a model for drifting assemblies, which may, for example, represent memory. And this assembly behaves similar to a thread. At each time, the assembly consists of a different ensemble of neurons. But because the change is gradual, the the input and output neurons are able to follow the change of the assembly. This is similar to a thread where, which consists of different fibers at each location, but due to the overlap of the fibers, we have a nice and continuous thread which we can follow. And because of the following of the input and output neurons, we have the conserved behavior. Also, this has recently been published. If you're interested, you can read more details and analysis and further analytical models and so on there. So finally, let me thank the entire neural network dynamics and computation group at the University of Bonn, the Institute of Zoology and the Institute of Genetics for a lot of support and very fruitful discussions. We are indebted for funding to the Bernstein Network and to the German Research Council. And of course, I would like to thank you very much for your attention.
Thank you, Raoul, for the wonderful talk. I already see that Rafa has a question.
Yeah. Raoul, so question about the first part of your work. Both parts were really exciting and beautiful. Just looking at the hydrodynamics, the vertices of the water, I thought that to me, that was some of the most fascinating results that you have, because it could imply that the way that this jellyfish is computing to perform a behavior is also by using the physics of the water as a computational device. So in that way, for cnidarians that have so few neurons and so simple nervous system, so in a way, I mean, as bilaterians, everything we do has to do with our muscles, but maybe for cnidarians, a lot of what they do doesn't have to do with their muscle, has to do with smartly manipulating the water, no?
Yes, this is a very good point, a very interesting point. Indeed, one thinks that cnidocysts, which are all over the body, may have axons connecting to the nervous systems. I mean, that's the thought. And indeed, they could feel all this water and all this vorticity around. Yeah, a very good point. Yeah.
Thank you.
There's one more question from an anonymous attendee. It says, what's the drift mechanism in the model you presented and what's causing the drift?
In the second part, yeah. Yeah, so what is causing the drift? So what is happening here is that the neurons are usually they stick to their assembly, but from time to time they randomly co-fire with another assembly just because of this irregular background activity. And then the weight to and from the other assembly are a little bit increased. And if then by chance this happens a few times more, or by chance the neuron fails to spike together with its own assembly, then the connections to the original assembly are weakened and the connections to the new assembly are strengthened, and the neuron then quickly makes a quick transition to the other assembly.
Thank you, Raoul. There's a few more questions in the chat. I suggest you answer by typing. I suggest we move to our next session, and Rosa is going to chair the next session. Rosa, please go ahead.
Thank you.
Okay. Hello, everyone. So it's a great pleasure to chair this last session. So we will move from leave the fascinating world of cnidarians and move to more, I don't know if I can say that, classical lab species, and hear about ensembles, first in the context of navigation, at least for the first two talks. And so the first speaker of this last session is Vivek Jayaraman, who is group leader at Janelia and also the head of mechanistic cognitive neuroscience there. And he made a great seminal contribution to understanding how the causal links between neuronal activity dynamics and decision in the fly. So Vivek, the stage is yours. Thanks.
All right, I assume you can hear me. and hopefully you can see my slides. Is that true? Yes, thank you, Rafa. All right, thank you, Rosa. Thank you, Emre, Rafa, for the invitation to come talk here. Always nicer in person, but I'll do my best with the virtual format. All right, so I thought in the interest of kind of provoking things a little bit, I was going to kind of emphasize what I think is the importance of cell types, however you define them, kind of functionally or morphologically or genetically, and connectivity in trying to understand how neural ensembles perform computations more generally. It's not just a fly thing, I think, in general. So before I get into that, let's talk a little bit about what internal representations I'm going to be referring to. I'm talking about navigational representations. So as many of you know, if you happen to record from many regions in the rodent brain or in the mammalian brain more generally, you find cells like grid cells or head direction cells or place cells and the like. And it turns out that if you, with some tweaking, train an agent, an artificial agent, to perform various kinds of navigational behaviors, flexible navigational behaviors. By flexibility, I mean it's not completely dependent on exactly the same setup. Some cues could be missing and you still have to find your way to the goal, etc. If you train on those kinds of tasks where there's some involvement of memory and requirement of retaining some sense of the structure of the environment and your place in it, well, sure enough, you find representations similar to these. And so typically you'd say these are representations found in densely recurrent circuits, typically in the middle of the brain, because a lot of processing needs to happen before you get to these. And further, you might say, well, okay, maybe one way to try and understand the activity patterns that one gets in, let's say, different areas of cortex or thalamic areas or whatever, one way to say this is to say, well, if I look at the activity patterns in different parts of a network, and hey, I find a deep network that is trained based on some particular learning rule and some objective function, and I find similar representations in certain parts of this network, that in itself is significant in terms of understanding something about the network. And I'd say there's some truth to that, even from my point of view. There is again an understanding of some sort there. However, sometimes there's this tendency to think, well, that's it. I mean, you kind of have understood what you need to understand. You've figured out you have representations. You have the learning rules and objective functions needed to generate those in the context of a task. What more do you need? And so I think this is where maybe there's a bit of a departure for me, because I would say that you could also maybe get at computations. So how do you generate such representations? And it's I think a question that is not just important from a biological implementation standpoint, which of course it is. You kind of want to know how the system actually does it. But additionally, understanding how these things are generated gives you much more insight into what these representations are actually, like really how stable are they? How resistant to noise are they? Why are they resistant to noise? How do they acquire that property? Things like that. So you can start to delve into things like that. And so, of course, it's an endless kind of mission to get at the nature of computation. You could go all the way, as subcellular and so on and so forth. And I think those are all interesting different levels to approach the problem. We've barely scratched the surface of all that. But what I'm going to try and tell you is how we do try to get beneath just the representational layer to get at the computations. And of course, we start with a significant advantage. We start with smaller brained creatures, flies, and in this case, with serious genetic tools. So insects in general, of course, you probably know, are incredible navigators. They do, bees or ants, I don't need to tell you. Lots of people have worked out just fascinating behaviors there. But flies can do some fun things as well. So in this particular case, I'm just showing you a few examples of flexible navigation in the fly. So on the left here, you have an example from a collaboration between Michael Reiser, my colleague here, his lab, and Charles Zucker's lab. This grad student, Tyler Ofstad, showed something very similar to the Morris water maze. Essentially, you have one cool spot, which is in an otherwise heated environment, and there are visual cues on the walls around this little arena, and flies have to work out, there is this spot, it's kind of, the cues there indicate where it is, and so I should sort of hang out there as fast as I can when the floor heats up uncomfortably. And so, the trajectories that you get at first, the flies are all over the place. Over time, they get better and better until at some point, by let's say the 10th trial, which is just about 30 minutes in, flies get really good at moving from one spot to this new spot. I mean, this is the old spot to the new spot just in a direct path using the visual cues, and you can do a probe trial in the classic tradition, and indeed they hang out near where they should hang out based on the association with the visual environment. Similarly, a simplified version, a one-dimensional version, is something Martin Heisenberg's lab developed a while ago, where you have a visual environment with multiple cues in it, you punish a fly based on which cues it's looking at, and indeed you can train a fly to selectively spend time in the safe areas of this visual setting. And this is something we've replicated in the lab as well, and we have some ideas on how it's actually carried out. Finally, I'm going to mention one more example. In this example, which again has quite a bit of history now already, recent history, what flies are doing is, this is in darkness. They have one tiny spot of food in the middle of an arena. Initially, they kind of, they're hungry flies. They've been starved for 24 to 48 hours. They eat a little bit, but then they explore and they kind of go a little bit further out. And then as time goes on, they get further and further out, and they make these increasingly large loops away from there, always coming back in darkness. And you can do this experiment without, the standard olfactory controls. You can insert the controls in there, and they do it without those cues as well. So essentially, in all these cases, what I'm saying is that flies have the ability to navigate flexibly, and they have the ability to presumably rely on some kind of internal representation to keep track of where they are relative to some other thing without necessarily using a strong cue to do so. And so that sets up for us kind of the real problem of like, how does a network achieve this? Not just finding the representations, but how do you get to those? And the advantage we have is we have this lovely deep recurrent neural network, except it's kind of natural, and we have access to it thanks to a long, long history of just really beautiful electron microscopy work, none of which, by the way, involves us, that started in larvae, went to adult flies in different types of EM. But all this electron microscopy work has what they've given us in this most recent collaboration between the Fly EM team at Janelia and Google, who helped with the reconstructions, and our local proofreaders, is like a really high resolution view of what the synaptic connectivity of a central brain structure that we care a lot about called the central complex, what that connectivity is. And so 4 fabulous postdocs in my lab, Roma and Dan, who were then in my lab, and Brad and Hannah, who still are, basically spent a ton of time digging into this network and pulling out insights that I'll get to by the end of the talk. But this is something where we have access to the connectivity, so we get to play out these experiments of what if. Like, what if you knew the connectivity and you had some ideas of function? What could you infer from that? And that's the game I'm going to kind of play for you. But let's get back to this dense network. It turns out this wasn't just some random picture I threw on. It was a small piece of the Fly central complex network. These pinkish cells are actually EPG neurons. I might occasionally call them head direction cells or compass cells for reasons that will become very clear shortly. The blue neurons are called ring neurons. Among other things, they bring in multisensory cues into the compass system. So this sets up for us kind of the essential piece of the puzzle. You have a head direction representation somewhere, and you have access now to the connectivity from different, don't try to read this text. Even if you have a very large monitor, it's going to be hard. But these are different classes of ring neurons, these blue neurons, with synapses, synaptic strength, you could say roughly, onto the EPG neurons, the pink neurons. And so the EPG neurons are on this axis over here. And so each row is one ring neuron's weights onto these EPG neurons. And what you can hopefully quickly take away is from this block and this block, this is an all-to-all connectivity for most of these ring neurons. So most of these multisensory cues are being communicated to the compass system in an all-to-all network. Further, there's substructure within this. So if you look at different ring neurons, if you look at this ring neuron, ER4M, let's say, relative to itself, which is over here, you'll see, again, a dense matrix here, like a little block here. It's a block diagonal, but with exceptions. We'll come to those exceptions shortly. So essentially, the cues coming into this compass system, those cues, these are inhibitory neurons, and these neurons are essentially doing some kind of selection process just between them for the cues that make it to actually guide the compass. So you're in this world, you have various cues around you. Which ones should you tether to determine your bearings? That's the fundamental question that's being asked, that's being answered through this. But it's important to note, if I hadn't sorted these by type for you, this is what you would see. So there's structure here, but what really pops in the end is having some sense of what these different types are. You suddenly start to see fairly interesting things. Firstly, this all-to-all inhibition within a class of a ring neuron, which Hannah Haberkern realized could give you, this stimulus processing benefit of selection of important stimuli, salient stimuli. In addition, there's off-diagonal stuff. So for example, let's look at this ER1A has an off-diagonal, thick off-diagonal block here, which is telling you that one class of ring neuron can impact the activity of the second class of ring neuron. So you have ensembles, they have specialized roles, and different ones can have a priority in tethering to the world. Like, so you could have some cues that say polarized light or something like that, that is a dominant cue if available. But if the other cues are around and it isn't, you'll use the other cues to kind of figure out your bearings. That's something you can infer from the structure of this, assuming you have some idea of the cell types involved. Now, the nice thing is I have to show you some eye candy. You can actually see these neurons in the synapses in this EM image. So this is, the blue is the ring neuron. These are the pathways coming in from the optic lobe, from the fly eye. You can literally see the synapses here. And then you can see that the ring neurons make synapses onto the EPG neurons, the head direction cells that have projections here, and then they go up to this other structure. And there's a whole bunch of these ring neurons. So you're getting all-to-all connections from a wide variety of sensory cues into this compass system. And what you end up with is like this gorgeous matrix that you can look at. I mean, even from a structural standpoint, you can look at in this EM volume. All right, so this may make you think, okay, the fly has a hardwired set of connections between various cues. There's prioritization of some cues over others. Let's kind of dig into how hardwired it actually isn't. So an individual ring neuron, in this case, we're picking a visual neuron, but there's examples of other kinds of visual neurons that respond to polarized light, or in Totz's recordings in Rachel Wilson's lab to wind. But these neurons, the visual ones, have receptive fields that are actually spatiotemporal and focusing on the spatial aspect. You'll recognize these as sort of excitatory inhibitory subfields. And what they're picking up is visual features in the fly's surroundings. And so if you look at an individual compass neuron, that's this row over here, these would be the weights coming from different visual ring neurons onto that compass neuron. A different ring neuron might make a different weight onto that compass neuron. And as a whole, you get this matrix that I just showed you earlier of compass neurons that get synaptic input from a variety of different ring neurons with different weights. Except these weights are actually plastic, and there's flexibility in the system. So what this allows the fly to do then is take advantage of this flexibility to map different kinds of cues onto its compass system. So it's not like you can only function inside your room, although during these Zoom days, maybe that's what it feels like. But you can actually function in the outdoors too. You can try it, it still works. You go outside and you'll map those cues and you'll have your bearings, hopefully, you still have those. So the point is that the fly can kind of map any kind of different kind of setting onto its compass system. Something you can see here in this video, what you're looking at is Johannes recorded from these compass neurons, these EPG neurons, using GCamp in a fly that's walking on a ball and is in closed loop virtual reality settings where basically the cues are all around it. I'm just showing it to you on a flat rectangle, but it's all around. And there's a population of neurons. Each individual EPG neuron, I should have mentioned this earlier, just occupies one wedge, one little sector in this circular structure, the toroidal structure called the ellipsoid body. And once the animal gets going, you can see very clearly this activity actually tracks its orientation in this virtual world really pretty precisely. If we change the visual cue, the same thing happens. Basically, in a little bit, the fly maps those visual cues onto its compass using this all-to-all mapping and some kind of Hebbian inhibitory Hebbian plasticity, we think, mechanisms of which we're still figuring out. And then what you get again is a head direction bump. So essentially you get a single bump of activity that kind of goes all the way around. And it's important to emphasize, this is not just taking a bright visual stripe and putting, it's not like a photoreceptor array we're looking at. We're looking at an organization of activity into this, central bump that kind of moves around. Sang Soo, when he was in the lab, basically showed just how flexible this mapping really is. So he started off with this mapping. This is the normal mapping of, in this case, a flying fly, looking at a single stripe just for simplicity. When the stripe goes clockwise, the bump moves clockwise. And so the compass kind of tracks. He's imaging at very low intensity for reasons that will become very clear. So the signal isn't as sharp as in the previous experiments. He does a couple of minutes of this to verify, yeah, okay, this is what the mapping is. But then he asks, can you actually reverse the mapping? How flexible is this mapping from the ring neurons that map different kinds of visual features and sensory features onto the compass? How flexible is that mapping? Well, using optogenetics, he excites compass neurons in exactly the opposite order that they should be excited when the visual stripe moves around. And you'll see that here. So basically, the network wants to follow the stripe, but Sung-Soo won't let it because he's exciting with CS Crimson, a channel rhodopsin variant, and exciting it in open loop now in the reverse direction. Okay, like I said, the bump doesn't want to do that, but that is what he's forcing it to do. And then it's strong enough that over time, he can, in 5 minutes, flip the actual relationship so that now, essentially, it's like saying the fly is moving left, but thinking that it's moving right, because the world is moving in the opposite direction than it should from its point of view, because its internal representation is actually flipped. So it's a very flexible mapping between them. And part of the way that this mapping works out, and the reason for the flexibility and how the rules of how it works, we think is in a normal setting, this wouldn't be an optogenetic stimulus, it wouldn't be an inverted mapping, but it would be self-motion that actually drives the bump to a new place. And then you get visual cues or whatever other cues that happen to be coming onto the compass system at the same time as the bump is in a particular place, and you tweak the synapses based on that coincident activity. And what you then get is basically something that can map whatever sensory setting onto the compass. And indeed, that's kind of self-motion driven bump activity is what Johannes observed even when he was looking at walking flies way back when. And indeed here, the fly is in darkness, its eyes are painted black, and every time it rotates, you can see the kind of bump of activity rotate with it. And if it just stands there for a while, the bump of activity doesn't move away from that spot. The drift is very low. And most times the bump will resume activity or sometimes even maintain the activity over 30 seconds, 35 seconds from the exact spot that the fly last left it, or where it was left off. Okay, so of course, there's many models about this kind of activity. It's been studied a lot in the theory domain. What's exciting to us is that we've been able to maybe use this, the fact that this circuit is so well described anatomically, and that we have genetic tools from the vast majority of people who share these tools in the fly community. We can take advantage of those things and really figure out mechanistically how these the bump actually moves around in these cases, how this ensemble works, how the population dynamics arise. And so that's basically what we've done over the last several years. And work from a bunch of labs, including Gabby Mehman's lab most prominently also for this, has figured out, for example, how the bump representation updates when the fly turns. I'm going to, again, go into this in a little bit of depth, just so that I can make a certain point at the end. So in this particular case, I've been focusing on these EPG neurons, the ones that have arborizations in a sector of this ellipsoid body, and then they actually project to a different structure called the protocerebral bridge. The names don't matter. The point that matters is the place that this projects. There's a different set of neurons called the PEN neurons that come down from there back to this ellipsoid body structure, but with a phase shift. So neurons that might come from this glomerulus of the protocerebral bridge project one phase shifted slightly offset from where the EPGs that projected to that structure came from. And similarly on the other side, except the phase shift is to the opposite side. This goes all the way around the ellipsoid body. And so what you can easily see perhaps is if I have a little bump of activity that propagates to this structure in the protocerebral bridge, this place, and somehow I give a neuron that picks up this input from the EPG neurons, this head direction input, another bit of input from angular velocity neurons. Basically, let's say whenever the fly turns counterclockwise, what I can then do effectively is provide input back, that is heading dependent input and angular velocity dependent input to the side of where the activity originally was in the EPG neurons. And as long as I have this recurrent loop with this slight phase shift, I can move the bump from one direction to the other. And similarly on the other side, as long as the activity is conjunctively coding both head direction and angular velocity, I can shift it in the other direction as long as the anatomy cooperates, as Tanya Wolf showed beautifully it does. And so this is something that Jonathan Green and us both and my lab as well figured out is kind of the key to the puzzle of how maybe the bump moves around in darkness as well. And so the nice thing again with EM is you can actually see these neurons and the connectivity. I won't go all the way around in the interest of time, but what you can see is these are the EPG neurons. And if we take an individual one of these EPG neurons and we look at its partner PEN neuron, you can go from this EPG neuron up to its PEN neuron, and it comes back with a slight phase shift. You can see the synapses right there. And you can go all the way around like this. So the PEN neuron comes back, synapses onto an EPG neuron here, but where does it project? It projects back up to the neighboring glomerulus. And once again, there's a PEN neuron to match on the other side that will come down with a phase shift. And so you get this range of phase shift, the same phase shift all the way around, and you can essentially move the bump in one direction or the other, okay? And so we could verify that this was the case by patching onto the PEN neurons, knowing what those neurons were. In a walking fly, Stephanie Wigner realized they were indeed tuned to angular velocity. So if she recorded from one on one side, you got a tuning curve for negative angular velocity. If she recorded from the other, she got the other side. And indeed, they were conjunctively tuned to both angular velocity and heading. But what's important for me here is to emphasize knowing these types, knowing their connectivity really was the key to figuring out how the whole system worked together. You could speculate on it, but being able to actually demonstrate it required the kind of tools that we had access to. And so I do think there's a possibility to get underneath the representation, not just say it's conjunctive or mixed selectivity, but to actually understand why and how it is what it is. And so I'm going to finish up in actually, can I get a quick sense of time, Rosa? Do I have like 5 minutes, 10 minutes?
Yeah, you have, at least 5 more minutes.
All right, so I'll zip through this next bit a little bit, but this is where we're kind of going to use the connectivity alone to make inferences about what the function is. And in some cases, the inferences turn out independently to be actually the correct ones. So we figured that there's something remarkable going on in the structure. These phase shifts can be useful for a lot of things. And the particular bump being sinusoidal, as it turns out, is useful for a lot of things. And so there's long been this notion that the central complex might be involved in path integration. Uwe Homburg, I think, first mentioned it in 2004 in a paper that this could be what it's doing. And so, of course, everyone knows the ants do remarkably well at leaving from their nest, going off somewhere. And then when they want to go back, they make a direct path back, which for some reason is called a beeline. Well, no, bees path integrate too. And then, you can imagine how you would actually break down this kind of this behavior into an algorithm that a neural network could be carried out. If you say, well, okay, as long as you have some kind of vector computation where I can sum up these vectors and then reverse a vector and get back kind of the home vector, how to get back home, that would be great. Flies are doing some version of this, not exactly this, but some version of this. At least they're keeping track of their direction and distance to some extent when they perform these behaviors in darkness, where they come back home, not home in this case, but to a food spot. And so one lovely thing here is, well, what would be convenient to do these kinds of vector computations? David Turetsky and Reddish pointed this out ages ago. Well, it's kind of handy to have phasors. So something where you have a population activity vector that basically is organized into this kind of sinusoid that you then can sum and get back a sinusoid. You can do lots of operations on it and get back a sinusoid. It's kind of handy to work with these kinds of things if you're trying to represent vectors of this sort with these kinds of activity patterns. And so one really cool thing we realized, and Gabby Mehman's lab also realized, is that there are these particular patterns in the synaptic connectivity between the EPG neurons and their downstream partners in this protocerebral bridge structure, a set of neurons called delta 7 neurons, that actually have this sinusoidal characteristics. So these are the weights from the EPG neurons onto the delta 7 neurons in different columns. So different EPG neurons arborize in different columns. This is different glomeruli. This is what I showed you earlier. A bunch of them, you get something that looks like this. If I just put this, if I basically convert this into a matrix where I take the central part of this column and I just rearrange all the others, what I get back is essentially a sinusoid. It's very well fit by a cosine, as Dan showed. And so essentially, this network, through the combination of inhibition and excitation, is forcing this bump of activity to be sort of vaguely sinusoidal. And that, it turns out, could be good for many things. So we noticed that there are anatomical phase shifts of the sort I told you about in the ellipsoid body, this little shift in the projections, not just in the ellipsoid body, but also in a different downstream structure of the protocerebral bridge called the fan-shaped body. So if you were to project the phases of activity in that 360-degree circle all the way through the system, what you get back is neurons that have a 0 phase shift. That basically means that projections from two sides that have the same phase go to the same place, just like here. Or it has phase shifts that are offset. So these are neurons that are phase shifted by just a little bit because they might come back to one side or the other. Sorry, I missed that here. So these are neurons that might come back to one side or the other from their original place, as opposed to these ones that kind of coincide in their outputs in the fan-shaped body. What this gives you is the ability to do vector computations more broadly. You can have one large vector and one small vector, and you get back the resultant vector, just based on the fact that there's low activity here, high activity here, and you take the bump and you kind of squish it, and you get back a new bump that's also a sinusoid that's just shifted from where it originally used to be. Useful if you want to do all kinds of coordinate transformations, including ones that have to do with converting head angles to body angles. And so what Brad and the lab did was take this approach and propagate it through the fan-shaped body structure and find neurons with these very particular phase shifts good enough that you could construct from that essentially a 4-vector basis set. So the fly could, in principle, have a basis set for arbitrary vector computations. And this indeed is what turns out to be the case. So we speculated in our electron microscopy-based analysis that it might be useful for computing things like traveling direction and so on and so forth, like just all kinds of computations that have to do with taking in head direction input, using head and body angles, combining that with like input that tells you which direction the world is moving and getting your traveling direction, something that would be handy for even things like path integration later. Well, it turns out in independent work, parallel work carried out in Gabby Mehman's lab and also in Rachel Wilson's lab, that is indeed what these neurons seem to be able to do in this particular brain structure. Finally, in addition to all the things I've told you, from the phase shifts, based on the projection patterns to premotor areas, we can also start looking at how goal heading driven behavior might be encoded. Learning dependent flexible behavior, but dependent on whatever the context is, you kind of learn different kinds of goal headings. And in that same phase shifted way, you can kind of crisply capture, the actions you want to perform based on the past experiences you've had. And so I'll just finish up by saying, in all these cases, what we're relying on is combining some sense of function from recordings with connectivity and cell type to extract kind of the nature of the computations underlying these representations and how they get used for the behavior. Finally, I'll just acknowledge my lab. It's been a ton of fun. Also past members, collaborators, theory and anatomy alike, and then the broader Janelia crew without whom we couldn't do any of this work. So thank you.
Thank you, Vivek, for a fantastic talk. We have a question from Emre.
Fantastic, really, Vivek. Maybe these are naive questions, but I mean, we're looking at a network where it has to have persistent activity, right? The head direction cells. You turn off the light, they will stay there. Yep. And then we are also looking. We're also looking at a network where we have to keep an eye on the time, because especially for these path integration, you have to know not just the direction of the vector, but also how long that vector has been utilized. Yep. So how do you see these two things going together within that sort of loops? Yeah, so there's clear examples already of neurons, individual neurons that seem to have this capacity to integrate from a certain time. So Vikram Vijayan in Gabby's lab has this rather lovely thing on the egg-laying circuitry, and you can kind of see the integration in a single cell. And so I see subcellular computations as being kind of key to that part of the puzzle. And I have, obviously we know about the clock system in many animals and particularly in the fly very much, but at these shorter time scales, how you keep track of time is something that, I don't think we have a clue yet how the fan-shaped body, say, does it in these different contexts. And we're not even at the point where we have a clue. We have clues, but we don't know how the path integration thing of just keeping track of the distance works either. We have hints, but we don't have more than that. So basically the short answer is we don't know. Thanks. Rafa.
Vivek, this is beautiful, and I found this sort of vector computation fascinating. So if you have these basis vectors, I understand intuitively how you can use it to remap and this could be very important for navigation. But more generally, what other computations could you do if you had a basis vector?
Right. So the way that we've been thinking about it, and we kind of speculate a little bit in our discussion, a lot in our discussion of this paper, but basically the way that we see it is it's a general purpose thing. For example, if the fly is facing this way, something happens back there, you want to reach with your arm. You have many legs in this case, and you want to reach somewhere there's all kinds of vector computation that we take for granted that happen to get, you know, the right joint angles, to know where to reach based on the fact that the head is rotated relative to the body. If I'm just using the eye coordinate system or the head coordinate system, I get one thing. As long as I can transfer it into the body thing, I get another. My initial position of where my legs is determines which direction I would do anything. So, you know, Gwyneth Card has this gorgeous behavior where basically in her behavioral assays, the flies escape when there's a looming stimulus that comes up to it. And we've discussed often that in the way that these flies react, if you give them a little bit of time and it's not just a very quick escape, they'll rearrange their legs so that they move away from the direction of the loom. But you kind of have to have the ability to know where your legs currently are, where you want to shift them in order to achieve a certain thing. So we see in all these premotor things as well, like the need for coordinate transformations. And obviously, you know, there's a long history of primate work in various coordinate systems, like coordinate system transformations and so on, that we think is relevant to the kinds of implementations we're seeing. We think that this is the kinds of things that those might be using as well.
Thank you.
Yeah, maybe I'll have one last question. I mean, we're running late. About the size of the anatomical size of the bump.
Yep.
When I look at your movies, it seems like something that it's pretty constant. And when you do the stimulation as well, like the spread of the stimulation, the evoked bump and the spontaneous bumps, they look quite similar. And so my question is, does it match also connectivity? Is there
so the one thing, yeah, so this is a really good question. And it's one of those things where, unfortunately, the link between excitatory connectivity and bump size is really tricky, partly because this is a densely recurrent circuit and there's many sources of inhibition and excitation in the circuit. So it's really hard to just say, based on the EPG to EPG connection, the head direction to head direction connection, this is what you'd expect for the bump width, because the inhibition scales with excitation. There's some inhibition that kind of doesn't. So it's been really, I mean, we would love to know exactly what, you know, the JE is, so to speak, for this network, but we don't know it like at that level. And maybe to answer the first part of your question, so the bump width is not exactly constant, but it's, you know, roughly, like if you look at the end to end, you'd say it's about 180 degrees and so on and so forth. But
the stimulation that is able to perturb the internal representation, does it have to have a specific size, or is it?
No, so as long as, I mean, we've, of course, it's limited by our ability to stimulate to a certain level, but as long as it's strong enough, it's going to overwhelm the bump, because we can really drive it hard optogenetically.
Okay, thanks. I think it's time to move to the second talk of the session. So it's my pleasure to introduce Lisa Giacomo, who is a professor at Stanford University and has been also analyzing the circuits involved in spatial navigation and in memory using electrophysiology, behavior, and computational modeling. Lisa, please.
Great, thanks. Can you see my slides okay?
Yes.
Okay,
perfect.
Great. All right, cool. Great, thanks for the introduction and thanks for the invitation to speak today. It's always really fun to be a part of these meetings that bring a lot of different speakers together. So my lab studies memory and navigation and your sense of space. And what I'm going to tell you about today is a series of projects in which we've tried to understand how learning and experience, what the animals learned about the environment and its experience, can influence our sense of space. So animals and humans have to be able to accurately navigate in order to solve a wide variety of different challenges. The real problem, though, or the real challenge to solving these problems is that in order the animal needs some kind of internal map of their external spatial world. And they need this to not only know where they're currently located, but to remember where they've been and to subsequently plan where they might need to go next. So how does the brain accomplish this? You've heard a little bit about it from Vivek already in the fly, but in the mammalian system over the last couple of decades, neurons in the medial entorhinal cortex, which is shown here in blue, have been found to provide a lot of the neural substrates that you need to build this internal map of the external world. The first of these are called grid cells, and these have been proposed to serve as a type of universal spatial metric system. This is an example of a grid cell shown here. What you're looking at is the bird's eye view of the activity pattern of the grid cell after this mouse has explored a 1 by 1 meter open arena, where blue colors indicate spatial positions where the cell wasn't active, and red and yellow colors indicate spatial positions where the cell was highly active. And you can see even just by eye that in many ways this pattern is reminiscent of a longitude and latitude coordinate system, only instead of a square grid covering a map, the unit of the grid is an equilateral triangle. Now, grid cells have two particular features that make them very well suited to encode an animal's position in space. The first is that different grid cells are active in different spatial locations. So for example, these two grid cells share the same underlying geometric structure, but they're active in slightly different positions in the world. And that means that no matter where the animal is in space, there's at least a subset of grid cells that are active. The second is that grid cells come in different spatial resolutions. So this is a sagittal slice of the mouse brain, and in dorsal entorhinal cortex, you find grid cells that have a distance between their firing nodes of about 30 centimeters. And as you move to more ventral portions of entorhinal cortex, the distance between these firing nodes grows to several meters. What that means is that you can combine the activity patterns of a relatively small number of grid cells with different spatial scales and really accurately decode the position of the animal in the environment. Grid cells coexist along several other cell types that are defined based on their firing patterns observed as animals navigate through space. There are border cells that are active near environmental boundaries, and object vector cells that fire at a particular distance and direction from objects in the world and are thought to provide information about landmarks and boundaries. There are head direction cells that fire at a high rate only when the animal faces one particular direction, for example, the northeast direction, and are thought to provide a type of neural compass. And there are speed cells that change their firing rate with the running speed of the animal that are thought to provide a type of speedometer. Together, these neurons are thought to support behaviors like path integration-based spatial navigation and provide input to place cells in the hippocampus that are active at one particular place in the environment and are thought to provide the neural substrates for autobiographical memory. So despite the really fascinating physiology of this system, the question that's become of interest to us is how this neural map might actually support memory and navigation. So initial experiments proposed a dissociation between how representations in the hippocampus versus the entorhinal cortex might support memory and navigation. And the idea was that entorhinal cortex might contain codes for spatial position, orientation, and running speed that are independent of what I'll call non-metric cues. So for example, the presence or absence of a reward. Whereas hippocampal place cells might contain codes for spatial position that are sensitive to both metric cues, like the geometry of the environment, as well as non-metric cues, like the absence or presence of a reward in the environment. This dissociation would then allow your brain, for example, to form a unique neural representation for 4 different concerts that all occur in the same concert hall due to changes in your hippocampal place cells. And yet your entorhinal cortex would allow you to have a universal spatial metric system across these 4 events because they all occur in the same context that would allow you to flexibly navigate through this space. However, work subsequent to the discovery of grid cells began to reveal that the entorhinal cortex may be more flexible than initially thought. So the first set of evidence in this vein came from studies that showed that entorhinal cortex codes are sensitive to changes in environmental geometry. So the basic paradigm showing this, it was first shown in rats, and then we later replicated it in mice. And so the basic paradigm is to have an animal explore a particular environment, in this case, a square, and become really familiar with this environmental geometry. You would then insert a barrier and have the animal explore this smaller portion of the environment after it had become familiar with this larger environment. And under these conditions, what you observe is that grid cells will selectively rescale along the axis that was modified. So in this case, for example, they will rescale along this modified axis, but they'll remain relatively unchanged along this unmodified or static axis. It's not just grid cells, though, that are sensitive to changes in environmental geometry. Speed cells actually also change their firing rate by running speed relationship under this paradigm. And this is an example of one speed cell that did that here. So what you can see is the firing rate of the cell on the y-axis and the running speed of the animal here on the x-axis. And you can see that between the baseline and modified conditions, the slope of this relationship is actually changing. A subset of head direction cells also shift their orientation to align with the modified axis. And this is an example of 2 head direction cells shown here that did that. You can see their tuning curves in the baseline environment in purple, and then their tuning curves in the modified environment in light blue. And you can see in this case, this cell reoriented towards the modified axis. And in this case, the cell actually increased its firing rate to align with the modified axis. So entorhinal codes are not only sensitive to environmental geometry, however, but also to changes in the animal's behavior. And in recent work, we considered entorhinal spatial maps when the animal was running fast versus when it was running slow. And we found that when the animal was running fast, more neurons in medial entorhinal cortex encoded information about the animal's position and head direction. So for example, here, a neuron when the animal was running slow may encode no information. And when the animal's running fast, it actually may start to encode information about the position or head direction of the animal. This change in coding was actually due to an increase in the sharpness of tuning. So this is an example of a position coding cell in which we've actually matched the spike rate and the occupancy. And you can see that when the animal's running fast, you actually get some significant tuning for this position here and maybe a little bit up here. But when the animal's running slow, you really don't see any specific tuning to any particular position. So these earlier works led us to thus ask, do entorhinal codes potentially adapt under other types of task conditions? So up until this point, the vast majority of studies examining entorhinal coding had employed just a single behavioral paradigm, which is foraging for randomly scattered food rewards in an open arena, like an open square box. However, many ethologically relevant navigational strategies don't rely exclusively on random foraging and instead often employ more complex behaviors like goal-directed navigation. And so we wanted to ask, are entorhinal codes potentially influenced by tasks that integrate a remembered reward location? And so to ask this question, we designed the following task. And we have a black box and a white box. And when the animal is in the black box, they're just going to randomly forage for food, sort of the oft-used paradigm that's used to look at entorhinal coding. And then when the animal's in the white box, it's going to alternate between 2 different types of behaviors. It's going to randomly forage for food. And then when it hears an auditory tone, it can navigate back to an unmarked reward location, remembered reward location, and it can receive a large food reward. So instead of receiving just kind of Cheerio dust, which is what we use for the random foraging, it actually receives an entire Cheerio. Critically, these 2 boxes are in the exact same spatial location in the room. So we literally just lift the black box up and put the white box down. And in addition, we compare all of our experimental data to a set of control animals that experience the black box, the white box, and the auditory tone, but they don't perform a task in this white box. They just randomly forage in both boxes. So what we found, to our surprise, was that in our experimental animals, many grid cells actually change significantly between these 2 environments. So for example, they might change their orientation, which is sort of the tilt of the grid pattern. This is an example of one grid cell that did that between environment 1 and environment 2. Or they might change their spacing. So for example, the spacing here for this cell gets a little bit smaller in environment 2 compared to environment 1. Grid cells weren't alone in these changes. Border cells also changed between these 2 environments in just our experimental animals. So here's a border cell, for example, that rotated from being active on the east wall to the north wall. And here's a border cell that sort of tightened up its tuning between the 2 environments. And head direction cells also change between the 2 environments. So this is an example of a cell that just shifted a little bit. And then in other head direction cells, we saw really significant shifts in their orientation tuning between the 2 boxes. We didn't see anything like this in our control animals. In our control animals, we saw highly stable grid, border, and head direction cells between the 2 environments. So what this tells us is that despite the fact that these environments have this underlying shared geometric structure, entorhinal maps are actually showing signatures of contextual remapping, the type of remapping we would really only expect to see if the animal went between 2 completely different contexts, like 2 completely different rooms with a change in task demands. Really interestingly, entorhinal maps appear to restructure to incorporate the remembered reward location. And this is just one way in which we looked at that. So what I'm showing you here is the distance of the animal from the reward zone on the x-axis and the normalized firing rate of all the grid cells that we recorded on the y-axis. where 0 here on the x-axis is the closest the animal can be to the reward, and 100 is the furthest they can be from the reward. And you can see that in environment 2, you actually get an increase in firing rate near the reward compared to far from the reward. And this is a larger, it's a higher firing rate than you see at the same position in environment 1. And this gray curve is just the difference between these red and blue curves. It turned out that this was because the relative activity of grid cells would increase near the reward zone in environment 2. So whatever grid nodes happen to be close to the reward zone would increase their firing rate, while the grid nodes further from the reward zone would either remain unchanged or sometimes even decrease their firing rate. These changes in firing rate then supported improved spatial decoding at the remembered reward location. So here on the y-axis, this is the error in our decoding. So this is how well we can decode the position of the animal in the environment. Smaller numbers mean we can better decode the position of the animal. And again, on the x-axis is the distance of the animal from the reward zone. And you can see that we can better decode the position of the animal near the reward zone compared to far from the reward zone in environment 2. And we can do this better than the same position in environment 1. And again, this is just the difference between those two curves. Okay, so I've just shown you that environmental geometry, the running speed of the animal, and a change in the task the animal is performing can all drive changes in the entorhinal spatial mapping system. But these previous works really all focus on the dynamic coding properties of single cells. So for example, looking at the tuning curve of this individual grid cell in a baseline versus a modified condition. But a lot of the computational models that are capable of generating things like grid cells describe entorhinal coding properties as emerging from recurrently connected networks of cells. And what that means is that the individual activity of any given cell in this network is not fully independent of the activity of its neighbors. And so we wanted to ask, to what degree do dynamic coding properties manifest at the network level? And some of the challenges in looking at this in some of our previous data sets were that the behavior is very variable. The animal rarely ever takes the same trajectory twice. These recording sessions are often temporally very short. They might be 20 or 30 minutes long, but most critically, we really only recorded from a small number of neurons simultaneously. We had one rat that was, around 60 neurons, but you really need a lot of neurons to look at population dynamics, especially when animals are performing these more complex behaviors. So to address these challenges, we turn to NeuroPixel's recordings in virtual environments. So virtual environments have the strength that they can force the animal to take the same trajectory hundreds of times, and you can record very long sessions while maintaining a really high behavioral output. NeuroPixel probes then allow us to record from almost the entire length of the mouse medial entorhinal cortex simultaneously. So this is a histology image, for example, of one of our NeuroPixel probe recordings. And this allows us then to record several hundred neurons at the same time. So this is an example of some of our first NeuroPixels work in which you can see the number of neurons that we recorded in entorhinal cortex along the x-axis here. And you can see that oftentimes we're recording from over 200 neurons and sometimes even over 300 neurons. So we decided to use this approach to ask whether or not entorhinal maps might show flexibility when the spatial environment, so the context and the task demands are invariant. And so again, we used this virtual reality setup and NeuroPixel's probe recording, and we had an animal explore a virtual environment for very long periods of time, but nothing changed. The animals just had to perform a random foraging task. So they would see a little visual cue in which they could slow down it in order to receive a water reward. And that location would change across individual trials. So there's no change in the task demands and there's no change in the sensory context. It's always the same environment. And under these conditions, we found that cells indeed have very beautiful spatially stable firing patterns. This is an example of 3 individual entorhinal cells that we recorded from. The position of the animal is here on the x-axis, and here on the top, these are different trials that are stacked along the y-axis. And you can see these individual dots indicate spikes of this particular unit. And this lower panel here is the average firing rate, the tuning curve of the cell over the course of this set of trials. And you can see that all 3 cells have these really nice, stable, periodic representations of the environment. But you might also notice that I'm actually only showing you 40 trials here, 260 to 300. And in fact, to our surprise, when we looked across the entire trial, so now this is 400 trials, what we found was that, yes, there were these very spatially stable maps, but there appeared to be multiple maps. And entorhinal cortex appeared to actually be sort of switching between these multiple maps over the course of this very long recording session. And so this told us initially that entorhinal neurons do dynamically vary their tuning properties despite this invariant sensory context. But to examine this from a more population perspective, what I just showed you was, still this single tuning curve level. We created these trial by trial similarity matrices. So all we're doing to create these is taking the activity of all of the neurons that we recorded on a given trial and correlating that with their activity on a different trial. And so these black sort of box representations here indicate trials in which all of the cells had a highly correlated representation. And then when you switch to a white box here, that means that these trials are all correlated with each other, but they're not correlated with this earlier set of trials. And so this checkerboard-like pattern that I'm showing you here indicates that entorhinal cortex is actually flip-flopping back and forth between two different spatial maps over the course of this recording session. This is a different session in a different mouse and a third session in yet another mouse. And again, you can see this underlying sort of checkerboard-like pattern in all three examples. But you might even be able to tell just by eye that the frequency and time between these map transitions is quite heterogeneous across mice and even across sessions. So even the same mouse might not show the same pattern across different sessions. And so this led us to ask, what factors are really contributing to this remapping? And based on our previous work where we had seen that the running speed of the animal can drive these changes in sort of the sharpness of tuning, we looked back at the running speed and locomotor behavior of the animals. And I'm just going to show you this first for an individual example session. So here on the top, this is the trial by trial similarity matrix. And then here on the bottom is the trial number on the x-axis and the running speed of the animal on the y-axis. And then every time we detect a map transition, there's a red dotted line plotted. And what you might even be able to see just by eye is that in many cases, right around this map transition, there's actually a substantial dip in the animal's running speed. There's one here and here, another here and here. And in fact, that's not just something that you can sort of qualitatively see. If you quantify the running speed of the animal in the trial just before a remapping event versus a trial where the map remains stable, there is actually a significant drop in the animal's running speed just before these remapping events. And it's not just one session and one animal. If we look at all of our animals that showed at least two different maps of space, we find that the running speed of the animal just before a remap event here on the y-axis versus the running speed of the animal during a stable block here on the x-axis is substantially lower just before these remap events. And so what this tells us then is that these population-wide remaps are actually preceded by slower running speeds. So to better understand how running speed might drive remapping, we collaborated with two theoretical neuroscientists at Stanford to characterize the geometry of the position coding. And in this slide, I just want to give you sort of a fundamental sense of what this analysis looked like. And then on the next slide, I'm going to tell you what this actually tells us about the data. So this class of computational models called attractor models are very good at generating grid cell representations. And this class of models would actually predict that the trajectory of the network activity should trace out a 1D ring manifold. And this is an illustration of what I mean by that here, where I've color-coded the spatial position of the animal along the linear track. And if we were to look at the neural activity of our cells in a lower dimensional space, what we would expect to see is some sort of ring manifold in which activity moves around this ring in correspondence with the animal's position on this track. And in fact, when we look at our experimental data, this is an example from part of one of our sessions. This is in fact what we see. And this is for what I'm going to call a single map. That just means that we're not looking at these multiple maps of space, but just one for the moment. And you'll see that there is indeed this nice ring manifold that represents the neural activity and corresponds to the position of the animal on the track. Really interestingly then, when we look at our full session where we have these two maps available, we see two ring manifolds that are actually very similar, almost kind of mirror representations of each other. And what we show is actually happening during these remapping events is that you have these two ring manifolds and the neural activity is actually just jumping between these two ring manifolds, which is what's giving you these very discrete remapping events that I showed you on the previous slides. Okay, so what does this actually then tell us about the data? Well, this analysis revealed two key features of the data. The first is that these manifolds are more aligned than we would expect by chance. And what that means is that the amount of sort of stretching, rotation, or manipulation we have to do to one of the ring manifolds to make it perfectly aligned with the second ring manifold is relatively minimal. That's this manifold misalignment metric here. And what that means is that any decoder in the brain that might be downstream from entorhinal cortex that's insensitive to translation along this single dimension will still be able to decode the position of the animal despite the fact that the system is remapping. And we actually showed this is the case in the data. I just don't have time to go into it today. The second thing that this analysis revealed is that when there is variability in the neural activity kind of off of this manifold, it's preferentially oriented in the dimension that separates these two manifolds. And what that means is that anything that increases the network variability or the neural variability can actually predispose the network to remap. And so based on these observations, we wanted to ask, do running speeds then correlate with an increase in network or neural variability? And so to do this, we calculated how close the neural activity is, this is data, to the midpoint between the two manifolds that we'd extracted using this analysis I showed you a couple slides ago. And what we found was that the distance of the neural activity to the midpoint, which is here on the y-axis, is actually much smaller when the animal's running slow compared to when the animal's running fast. So I'm showing this just for 2 individual sessions. And then this is another way to look at it. So here, this is, again, 2 individual sessions, and these are the slowest running speeds on the top row and the fastest running speeds on the bottom row. And you can see that this neural activity is actually getting closer to each other, closer to the center when the animal's running slow compared to when it's running fast. And so you can kind of envision it then at a high level like this, where when there are slow running speeds, the sort of energy barrier for the network to cross between these 2 different maps is lower. And so this predisposes the network to remap. Whereas when the animal's running fast, the energy barrier between these 2 representations is higher. And so it's harder for the animal to remap. So this intersection between behavioral state running, particularly running speed in this case, and entorhinal remapping was really surprising to us. So we next wanted to investigate whether this was unique to entorhinal cortex or might be reflective of remapping that occurs across different navigationally relevant cortical regions. And so we decided to consider this investigation initially in 2 different regions, primary visual cortex and retrosplenial cortex. And we again leveraged the high cell yield afforded by NeuroPixels probes and recorded from thousands of neurons in these regions while animals navigated down a linear track that was composed of 5 different visual landmarks. So consistent with our previous work, we observed a high degree of remapping in medial entorhinal cortex without any change to the task demands or the sensory context. What I'm showing you here is a little bit different from what I showed you before. All I've done here is sort of group our entorhinal sessions into times where we saw stable maps versus multiple maps. So blue for stable and red for remapping events. And you can see that there's still a substantial number of blocks or trial blocks in which we see a lot of these remapping events, even at baseline. However, V1 and retrosplenial cortex were shockingly stable at baseline. So we then performed a manipulation, which our previous work had shown often evokes really strong remapping in entorhinal cortex, with the idea that this might push V1 or retrosplenial cortex further towards a remapping paradigm. And so for this manipulation, what we did is we changed the gain. So we essentially create a mismatch between the locomotor cues the animal has learned to associate with the visual flow of the environment. So now essentially the animal has to travel further distances in order to get to the same visual features. And again, here we see that this actually increases the amount of remapping that we see in entorhinal cortex, consistent with some of our previous work. But even under these conditions, retrosplenial cortex and V1 remained very, very stable. And so this highlighted, as with our previous work, the really strong influence that locomotor cues have on entorhinal cortex. And so to further characterize the influence of locomotion and distance traveled on entorhinal cortex neurons, we recorded entorhinal neurons as mice navigated in complete darkness. And from these experiments, we were actually able to identify a subset of entorhinal neurons that encode the distance the animals run. And so this is an example of 4 different recording sessions where the distance the animals run is here on the x-axis, and then different neurons are stacked on top of each other along the y-axis. And what we found was that different entorhinal neurons actually beautifully coded for distance traveled along this track. And they actually did it in a very modular fashion, which is the same way that grid cells tend to encode space when there's actually visual cues available. And it increased along the dorsal ventral axis, which is the same thing that you actually see in grid cells for spatial coding. And here I'm just showing that in 2 different ways. And so this points to locomotion then as a sensory modality that really strongly innervates medial entorhinal coding. So just as a final note, I want to just very quickly come back to where I started this talk. I've shown you now that entorhinal cortex is actually quite sensitive to geometry, task demands, and behavioral state, particularly locomotion, and that changes in these factors can evoke entorhinal remapping. But why have remapping in both the hippocampus and entorhinal cortex? And if you have remapping in both, is there anything unique about what information each can provide across different contexts? And so to consider this question, we've again turned to the strength of neuropixels and their ability to record from hundreds of neurons simultaneously. And in work we presented at COSIGN recently, we found that while grid cells and landmark cells, like border cells, remap between different environments, they actually maintain their relative temporal relationships with each other. And this had been shown in a couple of different ways in freely moving behavior previously. So here, what I'm showing you is the spatial tuning curve of 4 different grid cells and 4 different landmark cells. And we recorded these in 2 different environments. So we would expect these cells to remap across these 2 environments. And you can see the tuning curve for the cells in environment 1 in black, and then in environment 2 in either blue or red. And you can see even just by eye that across these 2 environments, these cells have very, very different spatial firing patterns. But if we look at the correlations between grid pairs across environment 1 and environment 2, they actually really strongly maintain their relationships. And in fact, if we look at grid landmark pairs across environment 1 and environment 2, they also very strongly maintain their relationships. So this is quite different than from what you see in hippocampal place cells, where, for example, 2 different place cells will remap between environment 1 and environment 2, but they won't really maintain any relationship relative to each other across these 2 environments. Whereas grid cells, for example, will also remap between environment 2, environment 1, and environment 2, but these 2 grid cells will actually maintain their relationships with each other in the temporal domain. Okay, so just to summarize quickly, I showed you that entorhinal maps are sensitive to environmental geometry and running speed, and that remembered reward locations can serve as a cue capable of evoking different entorhinal maps of space, and that entorhinal maps at the population level restructure in response to state transitions. And at a high level, we kind of envision some of the remapping we see in entorhinal cortex like this, where you could have a mouse that's very familiar with this particular environment and it finds random food sources throughout this environment. But one day there's, a really large pizza party over at this particular position. And so it might actually benefit the animal to create a new map of that same spatial environment with more information potentially even about this particular rewarded location relative to other positions in the environment. And then finally, I showed you that entorhinal maps may actually complement hippocampal maps. And so one question that we're sort of interested in thinking about is whether or not these preserved cell-cell correlations could allow entorhinal cortex to provide some degree of generalization of information across contexts relative to the hippocampus. And finally, as you heard today, a lot of the neural substrates that I described to you have now been found in a wide variety of species, ranging from fish and flies to bats, mice, non-human and human primates. And we think that some of the dynamic coding properties that I described to you today might allow entorhinal cortex to alter its coding features to integrate the contextual features that are needed to really support the wide range of navigational strategies that these different species employ. And finally, I just want to thank the amazing lab, both current members of the lab, as well as alumni in the lab that participated in the work that I presented today, as well as a number of our really fantastic collaborators, and thank my funding sources and all of you for your attention.
Thank you, Lisa. This was fascinating. So there is one question from Rafa.
Hi, Lisa. I'm still trying to get into my head the idea that the same neuron can participate simultaneously in several maps at the same time. But but how do you switch? What is the mechanism in which you switch from one map to the other map in a split second? How can you do that?
Yeah, so so if you have multiple attractor manifolds, you can that would give you this like rapid, or would predict that you would see this rapid and discrete switching between the two representations. We're still trying to understand exactly what the mechanism is. We think it's related to locomotion or acceleration or some kind of behavioral state change of the animal, but we haven't actually pinned down the precise mechanism that's driving these map switches. It seems to correlate very strongly with changes in locomotion and potentially acceleration, but we haven't pinned down the precise mechanism yet. It's a great question.
Yeah, I think of it just in terms of the actual physical circuit. So
I see.
So would it be like maybe an interneurons kick in and they disable one part and enable another one, or it's like I don't know, how would you visualize that? Because it's of course a multidimensional problem, but I'm more like a nuts and bolts kind of person.
I see what you're asking. Yeah, no, that's a great question. I don't think it's well known from like the precise like wiring diagram how it might happen. Typically, like most of the models will be able to get remapping by taking the same like two-dimensional attractor manifold and then just taking different slices across that manifold. But then what has to actually change in terms of like neural activity or neural circuitry to get that realignment is not totally known. I mean, you could envision, for example, that you have like a landmark input that comes in that basically, like what Vivek was showing, that, if you imagine a ring attractor, then you can have like a landmark that might change that could kind of re-index the whole ring attractor. And that would give you a type of what I'm sort of describing as remapping. So that's one way in which it could happen, but there could be other kind of like mechanistic circuit ways in which it could happen.
So you probably have the data from interneurons, like have you sorted interneurons from your NeuroPixels recordings, and have you looked at their behavior during the transitions?
Yeah, that's a great question. We do have putative interneurons, that we're identifying just based on firing rate. I don't think that we've looked at them very closely in the remapping. I think at one point we looked a little bit. We don't get nearly as many as we do of excitatory cells. That's kind of like the classic, electrophysiology challenge. But that's a great suggestion. We have enough data that we could certainly pool all of our interneurons and take a look to see if there's any kind of change. This type of remapping tends to be highly coherent across the population. So, almost all of the neurons are participating to some degree in it. So I would suspect we probably would see some changes in the interneurons as well.
So I see there are many questions in the chat. Maybe I'll just pick the first one and then we have to move to the last talk.
Okay, I can also answer them, I think, after, yeah.
That's just this one. Regarding the evidence for limited remapping in V1, did you find place cells in V1 responding independent of the visual cues? And how much experience did the animals have with the environment before these measurements were made?
Yeah, so most of the V1 neurons were responding to the towers in the environment. So most of them fired like at a particular distance from the tower. So we didn't see very many like V1 neurons that were spatially tuned that weren't tethered to the visual cues. And the animals had what I would classify as a lot of experience. I think in the paper, there's a supplemental figure that actually quantifies how much experience they had for various regions and recordings. But these were not like naive animals. They had done, many training sessions, and they were quite familiar with the virtual environment before we did the recordings.
Unfortunately, we're running out of time. Looks like we've been running out of time even before we started. But I wanted to ask Yan Li to speak, give a few words. Yan Li, if you can turn on your camera. So Yan Li is actually she's a PhD. She did her PhD with Muming Pu at the Chinese Academy of Science in Shanghai, and then she worked for 6 years as a postdoc in Fred Cage's lab at the Salk. And then she worked at Stanford with Tom Sudoff for 2 years. So, I think, Yan Li, you should have been a speaker. but now she's a program officer program manager for the Chan Foundation, who's supported this meeting, sponsored this meeting. And Yan-Li, the floor is yours, so you can say a few words about what you're doing and your plans. Yeah.
Thank you, Rafa, for your nice introduction. So, and put together so exciting meetings. I was joining in and off while I was doing the other meetings. So it's really great pleasure and to meet everyone online. So hopeful. So yeah. So after the pandemic and have chance to meet people and in person. So I really appreciate and Rafa and provide the opportunity for me to introduce Tintiao and Christie Institute here. So our foundation is focused on support fundamental and translational research. So in the field related to the brain and mind. So we are very proud and to work with the Caltech scientists to establish Chan Institute for Neuroscience in Caltech. So our center director Dave Anderson just gave great talk and today I saw he just left and the meeting has probably other commitment. So we are very proud working with the Caltech scientists and in the past couple of years and see so many exciting research and was coming out and from Caltech. And we also built very strong relationship with Caltech over there. So besides fundamental and the basic research side, we also work with medical school and hospital. So in Shanghai, and we established Chan Institute for Translational Research, partner with Huashan Hospital, which are affiliated teaching hospital, with Fudan University and also Shanghai Mental Health Center as well. So during the pandemic, we provide more than $8 million COVID relief fund to universities and hospitals in the US to help our partners and friends overcome this very difficult time. And in the past couple of years, and I initiated and reach out to neuroscience societies and work with them very closely, and also including other conference organizations like Cold Spring Harbor conferences and Golden Conference and scientists like Rafa, and to support science communication and collaboration. So in the past couple of years, we have supported more than 50 conferences in our field. So I'm very glad and start from this year. So I will work with Rafa to support the conference organized by Columbia Neurotechnology Centers. And in this year, we also create a Chan Institute Science Writer Fellowship to encourage young scientists and our graduate students and postdoc attending conference and share their voice and opinions and in our communities. So I was thinking, this is just very brief and introductions about our institutions. And I will also shortly share my email contact and in the chat group. And if any of you are interested in learning more about our foundation and our program, just feel free and to contact me. So it really is nice to see everybody here and such great and discussion and activities.
Thank you, Yan-Li. It's not often that you find someone on the foundation side, on the other side, that it's an expert in neuroscience. You can have actually a very high bandwidth conversation with. So it's quite a privilege. Thank you for your support.
Yeah, thank you, Rafa. So this is true. So it's not traditional. So I guess science and the career track. And if any student or postdoc is interested and to learning more about my career and the choice, and so you're happy to contact me as well.
Wonderful. And with that, I think we're done. So thanks for all the attendees for listening to the meeting, for the speakers to generously donate their time for this. And I personally I found it extremely exciting. I think I'm going to go to bed thinking of this remapping. How the hell does that work? Anyway, but looking forward to more meetings in the future. We'll keep you posted. Thank you. Emery or Rosa, you want to say something from Norway or from France? Are you ready to go to bed?
Just thank you everybody for joining. This was a really fun discussion, and yeah, I loved it.
Thanks.
All right, thank you.